Hello, everyone. Good morning. It is a great honor to be here to share my experience. I'm Sunggyun Park, and one of co-founders and Chief Product Regulation Division Officer of Lunit. In my role at Lunit, I have been responsible for leading the product development, regulation, and clinical trials of medical AI software. So through this presentation, I will share the global regulatory experience and insights gained during the development of medical AI software. So since you might not be familiar with Lunit, I would like to briefly introduce Lunit in general. So we are a first generation medical AI company based in Seoul, South Korea, and branches in Boston and Amsterdam. We strive to be the global AI healthcare company. So we are a founding member of CancerX, and our AI was recognized and guided by WHO for TB screening from chest X-ray. Furthermore, we collaborate with various global partners, including Fujifilm and GE, aiming to successfully grow our revenue. Recently, Lunit has become the first AI selected into a national breast cancer screening program in NSW region of Australia. So as an industry frontrunner, our product was integrated in the Singapore National Healthcare AI platform, and Saudi's MOH 2030 Sandbox implements AI chest screening solution in 5 major hospitals. So to achieve this, we are focusing on 3 core competencies. The first is technology. So given our presence in the medical field, the performance of our products is obviously important to avoid harming patients. So therefore, we are constantly striving to create best-in-class AI based on large-scale medical data. The second is medical expertise. Since the problems addressed by Lunit falls within a specialized industry with complex workflows and environments, it is essential to have a precise understanding of this industry. Hence, we are collaborating with various medical experts to make the right products, and also we have published over 200 major peer-reviewed journals with 100 research medical experts. The final third is global business. Since the beginning of Lunit, we have always had global expansion in mind. The medical domain's problem we are tackling is not just a problem in Korea or other certain countries. We believe that the problem we are addressing could be applicable to all countries. Therefore, we have been aiming for a global business. So we focused on establishing a global sales channels through strong business partnerships. Currently, Lunit's products are being used at over 3,500 sites in more than 40 countries. Moreover, recently, we announced the plan to acquire Volpara, which has installation at over 2,000 sites in the United States. So building on this acquisition, we plan to pursue a more aggressive expansion in the US market. So based on these core competencies, our mission we aspire to achieve is to conquer cancer through AI. Through AI-powered cancer diagnosis and treatment, we aim to increase cancer survival rates. To achieve this, we have developed the Lunit Insight, a cancer diagnosis support solution that detects disease from X-ray images. Additionally, Lunitscope, another product lineup of Lunit, is an image-based biomarker for immunotherapy based on pathology images and other related information. But today, Luniscope is still under development, so the upcoming presentation will be focused on the Lunit Insight product called Lunit Insight CXR, Lunit Insight MMG, and Lunit Insight DBT. As Lunit Insight is a medical device, product registration is necessary for the target market of this product. We aim to become a global company, and due to our partners being spread worldwide, registration was required in multiple countries. So therefore, with the assistance of various agencies, Lunit Insight has obtained license from over 40 countries worldwide, including Korea, the United States, and Singapore. Additionally, it has been submitted to more countries and is under review. Recently, just 3 months ago, Lunit Insight DBT, which detects cancer regions from 3D mammography images, received FDA 510(k) clearance. So after getting approval, as mentioned earlier, Lunit continues to publish papers to prove the product value ourselves. For Lunit Insight CXR, which detects 10 abnormal findings from chest X-ray through AI, there were 5 papers before obtaining approval for pre-marketing trials. However, since then, we have constantly conducted post-market trials in collaboration with various research partners, and the total number of papers now stands at 83. Similarly, for Lunit Insight MMG, which classify cancer regions from breast images, numerous papers have been published continuously after obtaining approval. So currently, there are a total of 172 clinical evidence available for the Lunit Insight product. These papers primarily test the performance in various clinical environments and also include usage examples of real-world clinical workflows. Here is one example of our clinical trial. It was a retrospective clinical trial conducted at the Karolinska Institute. This institute in Switzerland directly compared 3 commercial artificial intelligence algorithms for independent assessment of screening mammograms. In this study, Lunit demonstrates the best performance compared to other algorithms. And also in the same study, they claim that radiologists can successfully triage up to 60% of the entire cases without human interpretation, which can reduce their workloads by more than half in mammogram interpretations. Furthermore, we aim to establish more robust evidence through prospective studies. Building upon previous retrospective studies, Lunit was selected for a prospective trials with the Karolinska Institute. So more than 50,000 women was enrolled in this study. And in a double-leading environment, one radiologist with our product demonstrated higher cancer detection rates and lower recall rates compared to 2 radiologists. So the double-leading guidance in Europe ensured the accuracy of interpretations as 2 radiologists review each case. But on the other hand, it imposed a significant workload on radiologists. This trial demonstrates how AI could be utilized in Europe's double-leading environment. Additionally, we conduct an RCT study in which subjects are randomly divided into control and experimental group for comparison on our chest X-ray AI product at Seoul National University Hospital. The A group, where radiologists lead with the AI product, detected more actionable long nodules and showed a higher detection rate of malignant nodules compared to the B group, where radiologists read alone. The greatest benefit of AI software lies in its ability to run from real-world use and experience, and its capability to improve its performance for patients. However, there are many limitations in pre-market clinical trials that digest the incorporation of these advantages. As described in this table, the population enrolled in the pre-market trials often is not enough to provide sufficient clinical evidence. Moreover, age, range, and periods are often too narrow to sufficiently represent real-world environments. So, to gain many more insights for the medical device product, post-marketing commitment is required. So here there are differences between pre-market and post-market trials. Compared to post-market trials, pre-market trials have limitations such as restricted environments or small sample sizes. On the other hand, post-market trials offer much greater flexibility in protocols and also provide numerous advantages in terms of data and cost. Reversing these benefits, real-world evidence can be obtained by these trials. Based on real-world data, companies can watch how the product works in the real world and also obtain its true value among various countries and workflows. To the customers, it can provide the user experience with AI in the market so that it gives a positive business impact by having a good user experience. In fact, regulatory authorities make great efforts to actively incorporate feedback from various stakeholders like us to reflect many aspects in regulation in line with the development of AI software. But based on my experience, I would like to share a few ideas to upgrade managing AI medical device based on clinical data. So while the expression for AI score, we call it abnormality score, can be ambiguous, but this score serves as an indicator of the confidence level of AI in detecting abnormalities. Lunit uses specific threshold to differentiate whether a case is normal or abnormal based on this AI score. It means that changes in the threshold can affect the sensitivity and specificity of AI performance. Typically, our customer or institutions aim to define the optimal threshold through their own retrospective trial to suit their needs. When healthcare institutes prioritize sensitivity, they may lower the threshold, whereas they can raise the threshold when they prefer higher specificity. Regarding regulations, providing more flexibility for adjusting thresholds could allow hospitals to use them in a more suitable manner. However, manufacturer must monitor its results and ensure the performance of whatever threshold is selected by the users. As I mentioned earlier, the post-market clinical trials are a good tool to find out the improvement points of the products. Based on real-world evidence, manufacturers might want to release the newest version of the product including AI performance enhancement. When product changes, having flexibility for quick and easy approval changes could help provide better user experience to customers. But for the US market, we are currently preparing for PCCP, Predetermined Change Control Plan. If we get clearance for it, we expect that this aspect can be addressed through it. But to us, PCCP is quite new, so we are running this system using a pre-submission procedure or others. But luckily, I will learn more about PCCP from the next sessions. Here is the rest. The data is the most important piece for AI development and verifications. Manufacturers try to find the efficient methods in order to facilitate the use and acceptance of clinical data. However, obtaining a suitable medical image for both clinical trials and developing deep learning is hard for everyone. So if government-level image source operation can be possible, it will be greatly helpful to the manufacturers. Therefore, the support for sourcing large quantities and high-quality data is necessary. Moreover, centralization of medical standard data for clinical trials also can be required to the manufacturers. Here is the last. So I will conclude my presentation here. So thank you for your attention.
Hi, everyone, and thank you to Dr. Park for giving me my lead-in for Predetermined Change Control Plan. We did not plan that. I am very excited today to talk about Predetermined Change Control Plan, or PCCP. What I'm going to do first is give you a little bit of why I care about PCCP, why I think that this is a great benefit to many regulatory frameworks. And then I'm going to go into a case study. Medtronic actually had a PCCP for one of our products and show you what the process looks like. But before I get there, I want to just give a great thank you to MFDS and US FDA for this great symposium. It has truly been an honor to be here, getting to experience the entertainment with the dancing and singing to the wonderful food. I think I'm going to roll myself home from getting to enjoy all this Korean food, to getting to experience Korea for the first time. So just thank you, and most importantly, for this amazing discussion, bringing together these brilliant minds together in one room, and getting to learn from so many of you. So thank you to all of you. With that, I wanted to sort of first set the stage. We've heard from so many of these speakers about the great benefits that we see for AI in healthcare. We're seeing it today. We're used to talk about the future of AI in healthcare, and it really is here today. We've also heard from the speakers and about the importance of developing AI and technology responsibly. And to that end, Medtronic has put out their own AI compass to really confirm our commitment to apply AI to our technology ethically and responsibly, and where it will make an impact to patient healthcare outcomes. And so why do we care so much about AI in healthcare? We at Medtronic sort of really look at being at this pivot point today in the healthcare system. We see clinicians being overburdened with so much right now, aging populations in many countries, bringing many complex diseases. Also, in many countries, we're seeing a lot of staff shortages in the healthcare system. And then on top of that, we are all seeing this huge explosion of data. Data coming from our electronic health records, from our smart devices, and from many connected devices. And that is where we see AI and the potential of AI to bring so much benefit to healthcare, to really assist our physicians, to be able to look at this data and curate it in a meaningful and actionable way for our physicians, and to really create a more personalized experience for us as patients. And so I'm here sort of to talk about the evolving regulatory framework. I think one thing that I'll say is, we're seeing AI impact our products across all sectors. I think one thing that we're lucky about in the medical device sector is that we already have this very robust regulatory framework that has been in place for years and is very a flexible framework. And we see a regulatory framework being in place for everything from surgical tools to pacemakers, to mobile apps, to robotics, and now to AI. And what this regulatory framework really offers is this flexible framework that's risk-based, based on intended use, and ensures the safety and effectiveness of our products. And so really when we look at our regulatory framework and we look at AI devices, we're not starting new in most places. We're starting from this really robust framework. And what we need to look at is what is different about AI? What is unique? Where are the places that we need to evolve the framework to meet the needs of AI, to ensure that we're encouraging innovation while at the same time still ensuring the safety and effectiveness of these products? And that leads me to what I'm talking about today, predetermined change control plan. Because one of the things that we see in many regulatory frameworks is it's not built for the change control that AI offers. We know that from all these speakers that we've already heard, that one of the very unique opportunities about AI is that as we get more and new data, AI-enabled products can iterate more quickly to increase the performance. As we also know, many of our regulatory frameworks were built around hardware devices like pacemakers, that in the past, you would go to the regulator, you would get your pre-market submission, and then maybe you would wait a year or two before you go back to the regulator for your new generation. Well, AI is different. As we know, it can iterate very quickly with new data. And so we needed a mechanism for many regulatory frameworks to deal with AI to encourage its innovation while still ensuring the safety and effectiveness of the products. And so what is PCCP? Here is sort of the very simple explanation of what PCCP is. Really, it's this mechanism that in the pre-market submission to provide to the regulator your pre-market submission, like you always do, but with it, specific modifications that you plan to make post-market. And that's important, and I'll get back to that. It's really, it's not a whole bunch of random modifications, but really these specific modifications that you plan to make. Also with it, you have a protocol that describes how you're going to make those modifications. How are you going to do the development and the validation of those modifications? And then lastly, the impact assessment. What are the benefit and risk considerations of this, and what are the risk controls in place? I want to pause here for a second because next I'm going to go into our case study that really looks at the US framework. But I also want to recognize that PCCP is not just US based. There are regulators around the world that are looking into the PCCP framework. We've heard formally from some regulators like UK MHRA and Health Canada, in that they intend to utilize the PCCP framework. We also see in the draft EU AI Act some information about PCCP. We've also heard informally perhaps some future plans, perhaps with MFDS and other regulators in the room, of interest in utilizing the PCCP. And so one thing I'll note, and I'll bring this back at the end, a huge point around PCCP is that as we evolve in this process, it's going to be really important to have conferences like this to make sure that we're talking to each other and harmonizing these efforts around PCCP so that industry can utilize PCCP in an efficient manner. And most importantly, this is for the patients to ensure that we get efficient and safe and effective changes to the patient. So now I'm going to go into the case study with PCCP and Medtronic's experience with it. Just to sort of set the stage, really back in 2019, FDA put out their discussion paper to talk about this concept of PCCP. There was a recognition at the time that the framework in place was really built around these more hardware-based products. Like I said, these products that iterate more slowly. And so FDA put out this concept of the predetermined change control for the first time. They also put out their action plan related to that. And then there was this interim time where companies like Medtronic started to seize upon utilizing the PCCP. We went to FDA with our pre-submission, had discussion with them, and did our PCCP for a product, which I will talk about it. There started to be a lot of excitement around it. We could see that this actually worked. This actually had a great benefit of ensuring that we're doing this in a responsible way, in a safe and effective way. But once we get onto the market, to be able to iterate more quickly and be able to improve our performance. There was a lot of excitement, like I said, and also a recognition that PCCPs could be used more broadly. It didn't have to be just for AI. While we saw such great benefit for AI, it could be used for more broadly for software, for changes to sensors, or maybe even more broader than that, changes in sterilization or to help with shortages. And so legislation was passed in the US to say that PCCP could be used for all products, not just AI-based products. FDA then put out their draft guidance that's still out today in draft on AI-enabled PCCPs. FDA has also said that in the future, they're going to be putting out more draft guidance to talk about PCCP even broader than AI. And so we'll continue to learn. We'll continue to try to see the types of changes that can be made under PCCP. So now let's get to our case study here. We at Medtronic, we manufacture a lot of products. One of the products that we manufacture are insertable cardiac monitors, or ICMs. They are indicated for long-term monitoring of arrhythmias. At the time of this project, our business went out to clinicians to really understand what's the need in this area. And clinicians were talking about what I talked in the beginning of this overwhelming burden of increased data review. And part of the issue is that from the ICMs, they were getting a lot of false positives, of which atrial fibrillation and pause episodes were the most. With these false positives was leading to a lot more remote follow-up burden and also a lot more data review burden on behalf of the clinicians, and sometimes some unnecessary concern on behalf of the patients. So our business designs these two new AI algorithms to help adjudicate AF and pause episodes that were coming from our ICM. And really the idea was to preserve the high sensitivity, so to make sure that the true alerts were being passed on to the clinician, but to decrease the burden of these false positives, to decrease the AF and pause episodes. It's important to note that these algorithms are locked, so they are not continuously learning and adapting in real time. So here is what we submitted to FDA. We're going to focus on atrial fibrillation in this slide. As I noted, our ICM is inserted for many arrhythmias. Atrial fibrillation is one of them. And this was one of those arrhythmias that we were getting a lot of false positives being sent on to the clinicians. So you'll see what we tried to do is, or what we did, I should say, we preserved that high sensitivity that I talked about. We were at 99% sensitivity of the true positives being sent on to the clinician to make sure that they were getting the true alerts. But you'll see a great performance here. We had about 74% reduction in the false AF alerts being sent to the clinician. This was a great performance, but you can see that there's room for improvement here. There are still some, at the time, false positives being sent on to the clinician. And this is where we focused on the PCCP. What we wanted to do was ensure that as we got more data, once this device got on the market, that we could retrain this product to ensure that we could increase the performance more quickly. And this is what I talked about in the beginning of my speech of ensuring that we were able to do this more rapidly. And so we thought the PCCP concept that had been introduced by FDA was a great place for this product. So we went into FDA with our predetermined change control plan And basically, it boils down to this very robust document of what specific changes we want to make and how will it be validated. I'll note that this is a lot of work. Our business spent a lot of time. It's a very robust document because basically what it does is it shifts all that work that you would do post-market if you were going to go back to the regulator post-market onto the pre-market side. It is not for every device either, because some devices don't have their second generation, third generation already lined up. Some products you just want to get onto the market, and then you have sort of slower modifications, or you're still figuring out what your next generations are going to be. This product was prime for PCCP because we already knew what we wanted to do for our second generation and third generation. And so that's what we went into FDA with. We went in with a pre-submission. We had a lot of discussion with FDA, a lot of great interaction. And ultimately, we obtained FDA authorization both for the product and our authorized PCCP. So what did that mean post-market? We'll do a little drum roll here. We got onto the market, and what happened is that we maintained our sensitivity, 99%, but we had a great performance increase. We went from that 74% of decreasing those false positives to 88%. It was a great performance shift in a short time for our second generation as we were able to retrain with new data once we got onto the post-market. This is also a great example as we speak to the business of a place where maybe the second generation wouldn't have happened, or it would have happened a lot slower. And this has been a true benefit for our physicians and our patients. And you see this really high value of PCCP. The other thing that I want to just note, and I know some speakers spoke yesterday about the importance of AI with transparency, is that we really had this commitment to transparency, both in the pre-market stage before we even launched. We wanted to be able to understand sort of what the clinicians needed in terms of education and training to be able to trust these new algorithms. We wanted to make sure that we were giving them the right level of education, but not too much information to overburden them. And so we really set about sort of giving them that education so that they could gain trust of these algorithms, and also gave them the option if they wanted to be able to see the underlying data. And then with our PCCP and our submission to FDA, we also outlined what our transparency plan was going to be, our communication plan was going to be. And we followed many of the same course in our post-market stage as we did in pre-market to make sure that we're giving the right education at the right time when we're going to make the changes. And you'll see that when we make the changes, we notify users that a new version is going to come up. We give a description of the changes, the impact of performance, information about the clinician workflow and experience, and a reference to the updated manual. And then finally, I just want to sort of take a little time to talk about some of these key takeaways. As I noted, the really importance of PCCP are for those regulatory frameworks that are in place and that you need to go back to the regulator with post-market modifications. It is not for every regulatory framework, and I think that's important to emphasize. We're seeing really the need with sort of these established regulatory frameworks, like I said, that were built around these older medical devices that don't have the ability to iterate more quickly. And also it's for those specific modifications that you would have to go back to the regulator for, because some regulatory frameworks allow you to make changes without going back to the regulator. The other thing that I noted for our product, it was a locked algorithm. We have talked, though, about the future of adaptive algorithms that can learn in the field, and we see the possibility for predetermined change control plan for those adaptive algorithms as well. So these future great benefit. The other thing that I want to note is really this need for a harmonized regulatory approach. We are seeing a lot of excitement around predetermined change control plan and PCCP. But we really want to stress the importance of ensuring that we are all talking with each other to ensure that these approaches are harmonized, to make sure that we continue this efficiency, continue these, the ability to make these more rapid iterations in a harmonized, more global approach. We've also seen the benefit for PCCP for regulators and industry to decrease the amount of submissions. So it really helps with resources as well. It's a lot of effort, like I said, up front. However, we see that it reduces the amount of total submissions, so it helps with resources from the regulator point of view. But most importantly, it is a benefit for patients. And that is why we are all here today, to talk about the benefit of AI and healthcare for the benefit of our patients. Many of us in this room, including me, are patients ourselves. We see the excitement that AI has to offer, and PCCP is really a tool to enable us to increase the performance of our products more quickly, to get them more quickly into the hands of our patients, but ensuring that we are doing so in a responsible way to ensure that we continue the safety and effectiveness of our products. With that, I want to thank you. Thank you for your time today, and thank you again to MFDS and FDA.
First of all, thank you very much for the warm welcome, and I want to express my deep appreciation for our hosts throughout this week, Korea MFDS, in joint collaboration with FDA. It's been a very memorable experience, and it's truly an honor to be part of this. So for my talk, I'm going to set the stage with an introduction regarding a case study to frame the discussion, and then I'll go into some business opportunities that AbbVie is trying to leverage with the use of AI in drug development, and then discuss some of the specific applications and use cases in this area, and then also provide an overview of some of the regulatory considerations and challenges in this space. And then lastly, I want to discuss a really important topic about health misinformation as it relates to patients. So this case study example, you may already be aware of it with the IBM Watson AI system. It's rather dated. If you recall, back in 2011, IBM Watson was used to compete against two of the world's leading Jeopardy champions and was able to beat out both of them. And the IBM researchers wanted to see if the AI system could tackle even more complex challenges, such as in healthcare. And specifically, they wanted to see if they could leverage the experience of Memorial Sloan Kettering's Cancer Center program, the brain power of the researchers and oncologists there, to help patients globally by standardizing treatment recommendations and options in cancer care. However, as this system was being rolled out globally into other hospitals, it encountered some challenges. For example, at a hospital in Denmark, the oncologist there only agreed with the IBM Watson Health recommendations in 1 out of 3 cases. And it became apparent about some of the challenges that went into developing the IBM Watson system in this case. It was trained solely based on the doctors at Memorial Sloan Kettering Cancer Center and had a large emphasis on information from American studies. And a lot of the patient data that went into informing this model tended to be more affluent, which was a type of patient demographic being treated at Memorial Sloan. And so it became apparent that there was a disconnect between treatment practice as well as availability of medicines in different hospitals and regions throughout the world, as well as differences in insurance coverage, which made it challenging to abide by the recommendations with the system. And it had the potential for certain bias that was introduced. And there was a lack of explainability where a lot of doctors were confused about the underlying justification that led to the IBM Watson Health AI system's recommendations. And so I thought this is a nice case study example to look at some of the unforeseen risks and pitfalls with trying to use these technologies in healthcare. So AI/ML, what is it? And we've had really nice presentations throughout the week describing it. I want to provide an overview in very simple terms, what it does and how can it help, especially from a business perspective. It can help extract structured data and insights from content. It can help classify, predict, and transform data. It can help generate content from learned patterns in data. And then how can it help? It can help accelerate the work through intelligent automation. It can help scale capacity through duplication and continuity. And then it can help amplify and hone the quality and precision of work. This is an overview of the virtuous cycle for AI, and I'd like to thank our business technology team at AbbVie for framing this, the different value drivers in the use of AI technologies in driving value to the business. It's divided up into 3 different categories: enterprise, experience, and expert AI. For enterprise AI, just think of tools. Think of the way to drive greater operational efficiency and value in your business operations. Think of being able to automate administrative tasks, for example. And then if you look to experience AI, this involves people. This involves being able to drive meaningful insights from the users, whether it be healthcare providers, patients, and doctors throughout the process. And then lastly, data. With a focus on the data, you can have expert AI with greater analytical precision and being able to have better decision-making process throughout all of these cycles. And then throughout this, it's important to consider what are the areas of highest risk and how can we drive the most value while balancing patient risk, patient safety and risk. And what are the important safety checks that are needed throughout the way. And so AI/ML applications in the drug development life cycle, this is the traditional pipeline development model for drugs from discovery all the way through clinical development, authorization, and post-market. This is not meant to be comprehensive of all the different use cases for AI in drug development. But as we saw clearly from the discussions this past week, it is really important for the use of these tools to drive greater efficiencies in the drug discovery process, as we're able to better have drug target identification and screening and prioritization, but also in the clinical development space, which I'll discuss more later, about the use of AI/ML-enabled digital health technologies to collect data more seamlessly, and in certain cases in remote situations to enable decentralized clinical trials, and also the use of predictive models like digital twins to help streamline clinical development. And for AbbVie, the different AI/ML digital health technology use cases, we have an example of 3 different categories in image analysis, voice analysis, and motion analysis. These are just some of the examples that we are exploring. For example, in image analysis, being able to analyze skin diseases and conditions, especially for our immunology research, or even able to evaluate skin quality, features of texture and facial lines for our aesthetics portfolio, being able to understand the range of motion in inflammatory arthritis disease, and in voice analysis, trying to explore ways of doing voice biomarker recognition for understanding neurodegenerative disease. And then in motion analysis, being able to detect and quantify scratch for skin conditions, and even in bradykinesia and other movement disorders in Parkinson's disease. And so our data scientists, in this specific example, they were able to develop a deep learning model for measurement of psoriasis area severity index. This is also called a PASI score. And so briefly, psoriasis is a chronic inflammatory disease that causes painful skin damage and irritation. And the PASI score is normally measured through a trained clinician. That's a measure of disease severity for psoriasis. And our data scientists wanted to see if we could develop a deep learning model to more seamlessly measure this type of assessment. They looked at a historical clinical trial data of patients undergoing treatment for psoriasis, and they developed a training data set that included tracking of an individual's disease changes from baseline over time. At right are some examples of psoriasis area automated detection using the severity scoring system with this model. And so with this model performance, it was tested against the ground truth with physician-measured PASI scores. And we were able to demonstrate really close tracking in the trajectory of the physician scores from severe to clear skin with the predicted scores using this model. The graphs at right shows representative images from 6 different individuals over time with their PASI score over the course of their treatment. The dark blue represents the physician-measured score, and the light blue is the predicted score using the deep learning model. And so what this is able to demonstrate is the feasibility of using this automated deep learning model, imaging analysis technique to more seamlessly collect and analyze data, hopefully using it in remote clinical trial settings and decrease variability of the measurements over time. However, there are some regulatory considerations and challenges with the use of AI/ML-enabled digital health technologies. For example, being able to streamline and leverage them across different clinical development programs. This is just a very specific use case for measuring psoriasis using the PASI score. But how can we work with regulators to leverage the data collected from this use case for other therapy programs as well? And also in this discussion with digital health technologies, fit for purpose, DHTs based on context of use is really important. But we also have to consider, what are fit for purpose data sets? Fit for purpose data sets that are used as part of training and testing for these algorithms is also an important consideration. And then next I want to discuss prognostic models in clinical trials. These are often referred to as digital twins. Oftentimes there are important ethical considerations for allocating clinical trial participants to a non-treatment placebo group. And this is true for long-term studies as well as studies involving degenerative disease. And so there's the potential to use these prognostic models to decrease the number of patients that are allocated to these non-treatment groups. And so the patient digital twin prognostic model can be based on historical observational data, historical clinical trial data to develop the model. And it can use the patient's own baseline data to predict the progression of their disease if they had been allocated to the placebo group. And ultimately, it has the potential to not only decrease sample size, but also to improve the statistical power of the study and to help streamline the development program. However, there are some challenges with this approach, and it's still early days, but regulatory clarity on the approaches to using patient digital twins is needed, and we need alignment on the different terms in this space. Because I know digital twins is also a concept in manufacturing contexts. Digital twins are also used in non-clinical settings as well. And so gaining alignment on that is key. And then we also need regulatory guidance on the appropriate use of digital twins as it pertains to the contributions to the benefit risk assessment and regulatory decision making. And I know there's a lot of conversation that's active right now in the European space, especially with the EU Artificial Intelligence Act. But we are also exploring here in the United States about different ways of potential AI legislation. And I want to emphasize the importance of having a risk-based and flexible approach to AI regulation, with a particular focus of oversight, prioritizing it on FDA's existing health authorities, and then also acknowledging the importance of providing the support that's needed to regulators like FDA in building up their own AI infrastructure, their own expertise and capacity. I'm going to skip this slide for timing purposes, but I want to discuss potential regulatory frameworks for AI and drug development, thinking more globally. ICMRA, the International Coalition of Medicine's Regulatory Authorities, published a horizon scanning report looking at the different potential regulatory pathways with different AI use cases. And then, as mentioned by other speakers, FDA published their discussion paper on the use of AI/ML in drug development. There was also a companion diagnostic discussing this issue in the manufacturing setting. WHO published a document on regulatory considerations for AI in healthcare. And then EMA published a reflection paper discussing the use of AI in the medicinal product lifecycle. And I'll discuss in more detail on the EMA reflection paper and the FDA discussion paper. The FDA discussion paper provided a really thoughtful landscape analysis of the current and potential uses of AI/ML throughout the different drug development phases, from discovery to preclinical and clinical development. And it provided some important considerations on the use of AI/ML, and I appreciate FDA seeking stakeholder feedback as they develop their current thinking on this topic. The EMA reflection paper was rather similar, but I would say it was more guidance forward and guidance driven, and provided more specific recommendations as they're thinking about potential regulatory frameworks for AI in the medicinal product lifecycle. It discussed harnessing the potential of AI and how to adapt existing regulatory frameworks, developing guidelines and recommendations for best practice, as well as considerations for data management, governance, and statistical stringency. And it also discussed mitigating new risks, such as non-transparent model architectures, as well as data management issues, the potential for data leakage, model overfitting, as well as some of the unforeseen risks in this space. And so from both of these papers, I wanted to distill some of the key themes for these potential regulatory frameworks, the main ideas and main takeaway messages. First and foremost, patient safety should be the guiding principle, and we should have a thoughtful, human-centric approach to the use and development of these technologies. And then health authority scope. With limited resources, we should try to focus the oversight of these technologies in the drug development process on those that contribute to the risk-benefit assessment, on those that actually contribute to the regulatory decision-making process. And then, as we've discussed throughout this week, the importance of cooperation, international harmonization, and standardization is key. And we're experiencing challenges as different regions and jurisdictions start to develop their own regulatory frameworks and thinking on AI, as well as different pieces of legislation that begin to emerge, just causing more challenges with trying to harmonize different concepts. And then classification of AI/ML technologies as a medical device software, and the regulatory pathways needs to be clarified as pharma tries to leverage these tools and technologies for drug development. And then having a flexible and risk-based approach and understanding how to determine the level of risk based on context. And then trying to consider intellectual property protections, but it's a balance. It's a balance between public disclosure obligations with stakeholders, third parties, and health authorities, having that right balance of the information that needs to be shared. And this lends itself to transparency with different stakeholders from patients, healthcare providers, and health authorities What is the right level of documentation and detail that is needed for assurance of their safe and effective use? And then lastly, model development and monitoring is key, especially with guidance and considerations on training, on validation, on testing, on monitoring and model versioning moving forward. And then I would say in the digital health technology space, in the medical device software space, we can take a lot of important learnings from them. FDA published their final guidance on the use of digital health technologies for remote data acquisition and clinical trials, and it provided some really important regulatory flexibilities. For example, being able to leverage verification and validation activities in lieu of complying with full design control requirements for certain medical devices was really key. And then FDA's guidance on clinical decision support software. Although this was not AI focused, it brought up an important issue that we've been discussing this week on the risk of automation bias. And then with foundational guidance being developed through IMDRF, there's a newer document on software risk characterization that's out for public comment. And I think it provides some newer types of considerations when looking at software risk, medical device software risk. For example, considering the users, the use environment, the target population, even the clinical workflows and the level of automation that goes into these software technologies. And with AI/ML-enabled medical devices, how can we leverage and clarify the use of certain medical device regulatory frameworks for innovative drug development? We don't work in silos, and oftentimes in pharmaceutical development, we take elements from the medical device technology space to advance our programs. IMDRF developed their key terms and definitions guidance for ML-enabled medical devices. And so it's important to have a common lexicon from the medical device space, but also to the pharmaceutical space so that we're all speaking with the same language. And also with GMLPs is an important concept that some pharmaceutical companies are already adapting as this was developed initially with the 10 guiding principles and is now an active working group under IMDRF. And then with Cassie's talking about predetermined change control plans, it's really important about how to leverage these pathways to help streamline the management of certain changes over time and some of the life cycle challenges with these algorithms. And then lastly, I think this is a really interesting topic that's still evolving, the idea of technology certification. There was a recent publication in JAMA that proposed a nationwide network of health AI assurance labs. And the goal with this idea of technology certification through third parties is to be better able to evaluate and monitor the performance of AI models in healthcare settings. And then I wanted to discuss the potential risks of AI misinformation as it relates to patients, especially with the increasing use of these large language learning models as a primary source of information for patients and consumers. Dr. Califf was quoted as saying that medical misinformation is already having a negative impact on health outcomes, causing people to make plainly uninformed and adverse choices regarding their health. And so this statement from the FDA commissioner is really important for us to understand as we move forward with these technologies and manage the safety of our patients. Reagan-Udall published a nice report which showed that 8 in 10 Americans actively seek out health information every year, and a majority of them seek this through online sources. And with the rapid increase in these different types of LLM tools and chatbots, information from those sources is just going to accelerate. And so us as AbbVie, it's really important to have an importance on patient health literacy for our patients. Patients need accurate and up-to-date and trustworthy sources of medical and scientific benefit risk information. We also have to acknowledge that these technologies can accelerate access, especially for historically marginalized communities that have limited access to formal structures of healthcare. And at the same time, we need to be extremely thoughtful about cultural competence. There could be differences in language, in culture, and customs. As we saw with the IBM Watson Health case example, being able to develop a technology and rolling it out on a global level, you could have some unforeseen consequences if you're not thoughtful enough about some of the nuances in the difference in culture and practices in different areas. And then regulatory precedence and transparency and feedback is so important for us as innovators and manufacturers. FDA has done a great job forthcoming with establishing this public database of FDA cleared and approved AI/ML-enabled medical devices. And CDER has published some landscaping assessment reports providing information about the types of AI/ML technologies, tools, and informations that have been included in regulatory submissions. And then moving forward, how can we leverage these qualification pathways? How can we best engage early with FDA to obtain scientific advice on these programs that can take a considerable amount of resources to develop? And with that, I want to conclude by saying patient safety is the guiding principle. We should have a thoughtful, human-centric approach to AI/ML development. The use of these technologies can provide greater precision and efficiency and insights to help accelerate access of these novel therapies to patients. We need continued stakeholder engagement, such as these workshops and forums, in order to have greater transparency and dialogue across the aisle to discuss the idea about having a risk-based and flexible approach to these regulatory frameworks. And then also, we have to acknowledge that as companies build up our own AI capabilities, we have to support health authorities' resourcing and their capacity building, especially as they build up their own AI infrastructure and technical expertise so that they can achieve their public health mission for the greater good for all of our patients. And with that, I'd like to thank you so much for your attention.
Thank you. So I realize that my talk is going to be slightly different from the other talks, all the talks that have gone before. The other talks have been more in a broader scope, trying to be at a higher level, whereas my talk is going to be at about a very specific application of AI. But I hope that it actually will be a fruitful contribution to the wonderful discussions that we've been having in the last couple of days. So with that said, the background of my talk is similar to what Andrew Bate was talking about yesterday, is applying AI for regulatory purposes, so specifically in the post-marketing setting. So my daytime job is in using existing healthcare data, such as electronic health records and insurance claims data, to look at the effects of our products, both good and bad. But we should always realize that these data were not collected for the purpose of research. There are many issues, but one specific issue is that the outcomes that we're interested in are often not recorded in the way that we would like to for our research. And we actually need to extract these outcomes using what we call either operational case definitions or phenotype algorithms from the data. So such a phenotype algorithm is usually a set of codes combined with another set of codes that we'd like to see. So diagnose codes with procedure codes, for example, that we require to occur, and then when they occur, we call somebody a case. But of course, this is never perfect. We'll have outcome misclassifications. And that will affect the veracity of our research. And for that reason, the FDA in this document here recommends that we do chart review. So for people that are either cases or not cases, we should actually go back to the original charts, have a physician review those, and decide whether or not indeed somebody is a case or not. The recommendation is to do this for everybody in your study population. both cases and non-cases. But if that's infeasible because there are too many, then the recommendation is, well, at least take a sample of both cases and non-cases, so that you can compute your positive predictive value and your sensitivity, and do quantitative bias analysis to adjust your results. Now, in reality, the vast majority of studies that are being published don't even do that. What they do is they limit themselves to only those people that are identified as cases. They do chart review on a small sample of those. And as a result, you can only compute the positive predictive value. You can't compute sensitivity, and that limits you in what you can actually do with the results of such a case adjudication. Now, the reason why people do that is not because they're lazy, but because chart review is incredibly time-consuming and expensive. And so to solve this problem, we recently proposed what's called the Knowledge-Enhanced Electronic Profile the Knowledge-Enhanced Electronic Profile Review System, or KEEPR system, which will be the basis of the rest of my talk, so I have to talk about it, which is a system that allows you to systematize this chart review, and also allows you to do it on structured data, so both EHR and claims data. This is primarily the work of now my colleague Anna Ostropolitz, and this is her paper, so I highly recommend that you read that. So this KEEPR system follows three principles: adherence to clinical reasoning, standardization, and standardization is both in terms of input and output, because the system is designed to work against the OMOP common data model, which is a standard structure in which you can both load EHR data and insurance claims data. And thirdly, the principle of dimensionality reduction. And that last one is also very important because in chart review, often the problem is that there's just so much information about a patient, and only a very small part of that information is actually relevant to determining whether or not the patient is a case. Now, the way that KEEPR applies these general principles of diagnostics is it identifies a set of categories of information that we would like to have for our case adjudication. Clinical presentation, clinical plausibility, which falls into several categories, any diagnostics procedures that have occurred, treatment procedures and medications that are recorded, as well as follow-up care and any complications that might have occurred. We look at how the KEEPR system works in practice. You start with a cohort of people that you would like to adjudicate. So for example, you start with a cohort of patients that, according to your phenotype algorithm, have end-stage renal disease. Those go into the KEEPR system. You need to help this system a little bit by, for every disease that you want to adjudicate, you have to identify a set of concepts for every KEEPR category. So for example, for end-stage renal disease, one of the KEEPR categories is symptoms. And we might list the concepts of vomiting, edema, and dyspnea as concepts that might be symptoms of end-stage renal disease. Inside the KEEPR package, there are predefined time windows for every KEEPR category. So for example, symptoms, we only extract those from the 30 days prior to index date up to and including the index date. And with that information, KEEPR then goes to your database containing your patient-level data and extracts the relevant information, and then out comes basically a CSV table with a single row per person and one column per KEEPR category. So for example, for person one, we see that the symptoms that were recorded were vomiting and nausea 29 days before index, and dyspnea 11 days before index. So you have that for every person, every category, and that is what a physician can now use to adjudicate the patients. Now, Anna set up a whole experiment using the Columbia University electronic health record data with two human reviewers who used both the KEEPR system and full charts. A gold standard was established by a separate set of reviewers. If you want to know the full details of her study, please read the paper, but the conclusions are important in that she found that using this keeper system, the time to do reviews was more than cut in half, so that's a good thing. But of course also, but probably more importantly, we saw that there was no degradation in the accuracy as compared to this gold standard that was established. And in fact, it's not statistically significant, but those using the keeper system even got a slightly higher accuracy than those that didn't, probably because of the filtering of information. So it was easier to find relevant information because of the restriction to only relevant data items. All right, so that's all the background of where I want to go. Because of course the question now is, well, can we combine this approach with large language models? So here is the output of the keeper system for one specific patient. And what I've done is I've just simply taken the CSV file, one row, and I converted that into just a piece of text. So just every header of every keeper category, and then the information that was found as a textual representation, including the days that it was found in the patient's record. So the real question is, can we use large language models to review this information? Now, it's important to realize that, of course, this is patient-level data, so it cannot leave our organizational firewall. So we cannot send it to ChatGPT, but nowadays it's possible to have a wide variety of models running in-house. So we have available GPT 3.5 Turbo. But we didn't just want to use a fully commercial product. We also looked at the more easily available Llama 2. I'll show you in a minute that that actually didn't perform as well as we'd hoped out of the box. So we looked at another variant of Llama 2, a fine-tuning, which was at the top of the Hugging Face leaderboard at the time when we looked, called Sheepdog Llama 2, which was actually performing better, as I'll show. And so we ended up using that. But it's available on the same conditions as Llama 2, so you can just download it, and for almost all uses, it's free to use. Now, we created a training set, what I call a training set, but it's important to realize that this is not something that we used to actually train the model. We didn't fine-tune our model on this training set. This was a set that was constructed basically for me to do the prompt engineering to get to the system that we would then actually evaluate on the proper test sets. I think this is an important point to make. It's very easy, if you have a test set, to get really good performance on it by just keep on twiddling with your system until it works well on the set, but then you've lost all generalizability. So we created a separate set to do all the twiddling, and then we basically had one shot on goal for the actual evaluation. So what we did, we looked at 6 conditions, acute bronchitis, hyperlipidemia, hypoparathyroidism, osteoporosis, rheumatoid arthritis, and viral hepatitis A. We focused on quote-unquote hard cases. We found a total of 358 patients that we wanted to look at. We had one reviewer review these using the keeper system and the original data. And we used the Optum Clinformatics database, which is a US insurance claims database. So now for the prompt engineering part, so we had the text, the output from Keeper, again showing you here as directly coming out of the system, or at least transforming that CSV file to a text file. But of course we can't just give that to the LLM, we have to tell it what to do with this. And so that's what we did through the system prompt. And so we started with a very simple system prompt, basically just a yes-no query, act as a medical doctor reviewing a patient's healthcare data, determine whether the patient had the disease, summary, yes or no. And if we ask the LLM that together with the actual data from Keeper, we see that we get a very high sensitivity, 99%, but very poor specificity, 8.9%. Well, the first thing you learn about prompt engineering is you have to do a chain of thought prompting. So that's what we did as well. We asked the system to provide first all the evidence in favor and against the patient having the disease, and only then come to its conclusion. that reduced sensitivity a little bit, but specificity went up quite a bit, and the overall agreement went up. Continuing on this idea, we also asked the system to first write a medical narrative that fit the data that we provided it before it went on with its conclusion. That again increased sensitivity, reduced specificity by a little bit, but overall agreement went up, so we thought it was a good development. So now we started to look at, well, what is the system doing wrong? And so we took a sample of the failures and noted that the LLM was being a little bit naive in that it just always believed the diagnosis if it was present. So if on the day of the visit there was a diagnosis of the disease, the LLM would say, oh, there's a diagnosis, yeah, the patient has the disease. But anybody who's ever done chart review knows that that's not how it works. And so we had to explain this to the LLM. So we included this very large paragraph explaining that just the occurrence of a diagnosis is not enough. Could be there just because the person was being diagnosed or tested for the disease, and for insurance purposes, that code then had to occur, but it wasn't clear that the patient had the disease. And if we added that whole blob of text to the system prompt, now we see that specificity indeed goes up at almost no cost of sensitivity. And of course, overall agreement now is up by quite a bit. Again, we tried to see, well, what is the system doing wrong? We noted that the LLM didn't really know how to deal with any of the uncertainty that it saw. So sometimes, even when there was like an abundance of evidence, it would make up some story how it could be feasible that the patient didn't have the disease or the other way around. And so we added this this text that tried to explain it, well, if you think it's the most likely thing, then just say yes. And if you don't think it's the most likely thing, then say no. That again increased specificity by quite a bit. Sensitivity dropped a little bit, but overall agreement went up. The final thing we tried is what's called few-shot prompting. So we added some examples in the system prompt of the input and output that we were hoping to see. That actually didn't work very well. It did increase specificity, but the overall agreement went down. So as our final system, the one that we were going to evaluate, we went for the prior incarnation, so without the two examples. So I did all of that using the GPT 3.5 Turbo because it was by far the fastest. Then I started to evaluate that on Llama 2. And in the middle row, you can see that Llama 2 had high sensitivity, 99%, but very poor specificity, and overall agreement was not very good. And as I mentioned, we went to the top of the Hugging Face leaderboard, found Sheepdog Llama, and that actually has even better performance than the GPT 3.5. And so we went with that instead of the Llama 2. The point that I'd like to make here is that there are multiple good LLMs available, but you have to test them before you know which ones are good. So what does the whole system now look like? So here is the example system prompt with all the components that I discussed before. That is accompanied by the output of the keeper system. Here showing you a different patient who's more representative, who has more data points. And then here is the example response from Sheepdog Llama, where it goes into this clinical narrative, lists all the evidence in favor and against, and comes with a conclusion, in this case, correctly, that this patient is indeed a case. So we designed a total of 3 evaluation experiments. The first one was basically the same data that was used in the original Keeper experiment. So this was data from the Columbia University electronic health record system, about 4 diseases, with 2 human reviewers. The second experiment we did was using those same 4 diseases, but now using the US insurance claims data. And the 6th experiment used the same 6 diseases as we used in the training set. But now, instead of focusing on the hard cases, we just took a random sample, and again, using the US claims data. In all of the 3 experiments, we actually focused only on those people that were identified as being cases. So this would allow us only to compute positive predictive value, and later on I'll go into an example where we extend that. So here I'm showing you the agreement between human and LLM reviewers. So for example, the top left number is 83, meaning there was 83% agreement between Sheepdog Llama 2 using the Keeper system and the human reviewer 1 using full chart review. What you'll see already in this plot is that the humans actually agree with the humans just as much as the humans agree with the LLMs. I did a statistical test that there's at least no statistical difference. And you see that the medians are pretty much the same. If we split it up by disease, we see pretty much the same picture. What I find interesting in this picture is that actually reviewer 2 using the charts is the outlier, and he disagrees equally with the other human reviewers as with the LLMs. And so consistently even disagrees with himself using the Keeper system. But certainly there's nothing making the LLMs pop out relative to the humans. So here are the results from experiment 2. So this is using the same 4 diseases, but now the US insurance claims data. We see that the agreement is slightly lower overall, but again, there's no difference between human-to-human agreement and human-to-LLM agreement. And in fact, there's a slightly higher agreement between humans and Sheepdog Llama 2 as between humans and humans. Experiment 3, so the 6 diseases and again the US claims data, we see even slightly lower agreement, but again, no difference between humans and large language models. If we split it up by disease, we actually see something very interesting in that there is large heterogeneity across the different diseases. The middle column, hyperlipidemia and rheumatoid arthritis, there is strong agreement. Whereas for hepatitis A and bronchitis, bronchitis, there is quite a large amount of disagreement. Not different between humans and LLMs, but just overall disagreement. We believe that this is actually a data problem. We're using claims data, and the claims data do not contain all the lab values for all the patients. And so, for example, for hepatitis A, you would see that tests were ordered for multiple viral infections, but then if you don't see the results coming back, which one is positive, then you can't decide which one is, which is actually the correct diagnosis. So as I mentioned, these are all just on the actual cases. If we want to compute sensitivity, we need to have a much broader net, so we need to include also non-cases. One way that's proposed to do this is to use what's called a highly sensitive cohort. And so to illustrate that, we constructed a highly sensitive cohort for rheumatoid arthritis, meaning that any occurrence of a diagnosis, a symptom, a treatment, or a complication, or a lab test related to rheumatoid arthritis would be counted as being included in this highly sensitive cohort. So there's a very small chance that we would miss any RA cases, but probably with very low specificity. This turned up around 1 million persons, so that's even for LLM maybe too much. So we sampled 25,000 persons. We ran that through Keeper with GPT 3.5. That took about 40 hours, cost us $15. And in the end, only 527 of the 25,000 people, so that's 2.1%, was classified as an actual case by the LLM. Which is okay, but now we have an annotated set that we can use to evaluate existing or new phenotype algorithms. And so we took a phenotype algorithm from the Odyssey phenotype library, and we could now establish that it has a positive predictive value of 70.3% and a sensitivity of 79.1%. With confidence intervals that are not too wide, but I would like to have them narrower, just to make the point that 25,000 people gets you reasonable power, but you probably need even more. And doing case adjudication on more than 25,000 people manually is just not feasible nowadays. So depending on your view of LLMs and how much you trust them, I guess we can distinguish three use cases. One is just using the LLMs as co-pilots, so preparing a assessment for humans to then modify and review. If you were to fully put all your money on the LLMs, you might actually just use them to annotate all patients and have the LLMs decide which ones are cases and not cases and include those in your analysis. I think what we're proposing here is sort of a middle ground where we have just a sample of cases and non-cases. We use the LLMs to annotate that, and now we can compute PPV and sensitivity and use that for quantitative bias analysis. So, in conclusion, across all the three experiments that we did, we saw that LLMs agree with humans as much as humans agree with humans. Although, certainly for the claims data, we saw that the humans didn't put the bar very high. So that's actually something that we also need to have a look at. Certainly the LLMs have the advantage that they allow for scaling up, and that scaling up is important if we actually want to compute both sensitivity and positive predictive value and do quantitative bias analysis. We did notice that the LLM performance depended strongly on the choice of the system prompt as well as the choice of LLM. The zero-shot prompting that we used actually worked remarkably well. In theory, we could go much further. We could fine-tune these LLMs for this specific task, but that would require way more data than we currently have, a much larger training set. So I don't think that's feasible. So as we discussed, using LLMs for actual clinical care is something that we need to be very careful about and very need strong regulation. I think in this specific use case, the risk is somewhat lower because we're not misclassifying a patient in this setting won't directly cause harm. So I think it's a lower risk application. We still need to be careful, of course, but I think it's a very promising area. And with that, I'm at the end of my talk. I'd like to thank you for listening and my fellow researchers for their work. Thank you.
The mission of Rhino Health is to harness the power of federated computing as a way to improve healthcare outcomes by activating and federating healthcare data worldwide. As we know, healthcare data is today a very large asset, growing around 30% of the global the global volume of data is generated by healthcare. The CAGR of that, the compound annual growth rate of it is 36% and is the largest. And there's already over 50 petabytes of data produced annually by an average hospital. However, 97% of healthcare-related data is still unused, unreachable, and we still have a headroom of hundreds of billions of dollars in the US alone from being able to access more health data. As a result of this potential, there's been significant investments made over the last years, and there's some rapidly growing market segments, both in the space of healthcare analytics, population health management, and now AI in healthcare is a growing, already large and growing segment, as well as then healthcare interoperability solutions and real-world evidence solutions. In 2023 alone, there's been $25 billion investment in digital health, with a large part of it focused on data-rich products. In addition, in conjunction to that, regulators are pushing for more real-world data and real-world evidence in product development, both of medical device as well as drug products. The US FDA itself has published guidance about the use of real-world data in drug development. The Korean MFDS also recognizes real-world evidence as an adjunct to clinical trial data, and the European Union's EMA also released similar guidances. And today already a large majority of drug companies use real-world evidence with a potential to unlock hundreds of millions of dollars more per company. Okay, so far it sounds quite good, I think, and underscores the reason why healthcare data is such a rapidly growing industry. However, there's a problem with increasing volumes of data need to be shared. However, the main paradigm today of data centralization does not scale very well. It is associated with both high costs as well as a low value. Why high costs? Both because of a complicated regulatory landscape in which you need to work between HIPAA, GDPR, PIPL, and additional GDPR equivalencies, as well as different local versions of regulation. In addition to that, data centralization drives the need to create redundant data and inefficient use of existing compute infrastructure. You have research compute in the hospital, then research compute in the cloud, then research compute on the vendor side. In addition to that, there's a trust issue that drives complicated contracts, contract enforcement, et cetera, and as a result of that, a lot of the use of data today leads to low yield. The data accessible for researchers is often redacted and does not contain the full clinical experience of a patient. Most research is done on monomodal data rather than most AI, cutting-edge AI, which is done on multimodal data, as folks besides me today have presented. Data is typically not up to date and not rapidly refreshing, which also limits the use case of that data. You want to, for example, identify a patient for a clinical trial. Within 1 week of a clinical visit, that data becomes irrelevant since the patient has perhaps been gone through different lines of therapy, and a decision support tool would not be relevant to them anymore. And there is no linkage to the actual medical record. And so it's hard to action based on insights coming from medical data. The solution that we propose, and indeed is something that we see increasingly happening in the market, is the move for federated data technologies. We've already seen a strong push in moving to federated data, as in data that's mapped to common data models, with or without some central orchestrating mechanism. And in that, we've seen Project Odyssey's OMOP data model, Picori, HL7 Fire is to some extent related to a data model. We've also seen solutions coming out in the last 10 years for increasing federated analytics, meaning the ability to process one data silo at a time and then aggregate results, similar to companies like Trinetics being a prominent example of that. And then there's the new technology of federated computing that involves more sophisticated edge computing in order to process data as though it was sitting in one data silo and be able to work on AI-ready data. And I'll get into a bit of detail on what that means. In 2016, federated learning, which is the core enabling technology of federated computing, was proposed by Google. And by 2017, the tech world had already started leveraging that technology as for different non-healthcare users, such as mobile phone keyboards. And today, if somebody has, for example, autocorrect on their phone, the autocorrect doesn't isn't based on you sharing your text messages with Apple or Android or Google, etc., but rather you have an algorithm on your phone that gets smarter as you use your phone and contributes to a global AI model that is then refreshed on your device. Similar technologies to that also exist in autonomous vehicles, oil and gas, aerospace, etc. In 2019, the first implementation of this technology was brought into healthcare by NVIDIA that had the Clara FL original proof of concept, and I was part of the first real-world use of that technology between Mass General, Stanford, American College of Radiology, DASA, and a few others. And within a year, we conducted another experiment, which this time included 20 hospitals worldwide and included the actual federated learning as a software component installed in different data centers, meaning not on one kind of cloud in a virtual way, but actual data staying in different data silos around the world while being liberated in order to federate an algorithm. That was for a COVID prognostication model, which led to a publication in Nature Medicine and was kind of deemed the landmark of usage of federated learning on real-world data. In the same time, Project Melody also began in Europe, and within 3 years was completed and gave back results. In 2021, inspired by the exam study, Rhino Health, my company, was founded with the intent of scaling and popularizing this technology in healthcare organizations and life sciences industries worldwide as a tech platform. And today, by 2023, we are already rolled out to 40 sites worldwide. Our main focus is on healthcare organizations, biopharma, professional societies, and some health tech. This is the view of the Rhino network today. We are today at around 40 sites and expecting to get to around 100 by the end of 2024. As you can see, we have a lot of focus on North America, but also we are installed in KAIST, Asan, Seoul National University Hospital, and are rolling out to an additional 13 sites as part of a Korean research consortium. Federated computing enables the use of different methods in order to create new healthcare data applications that have not really managed to be built at scale to date. It allows to bring Gen AI to sensitive health data without requiring to move data from the hospital IT to a public cloud. It enables using multimodal data from different data sources. It enables to maintain a real-time link to source systems as the medical legal record at scale. It enables continuous monitoring and fine-tuning of AI performance. Some of the examples of already live applications of that technology today are the use of large-scale real-world data for regulatory review, for seeing how approved AI products function on different populations and different geographic settings across the US. It is to enable precision medicine and early detection of disease, such as the National Institute of Health and the National Cancer Institute in the US, early detection research network, and now with NRF as well for projects that's kicking off right now. It's also used by drug companies in order to create efficient data registries for safety and for early development, such as creating biomarkers, drug design consortia, and part of the use is also to be used by different drug companies collaborating with one another. such as using different docking data of drugs in order to predict drug properties and to actually design drugs from scratch. And it's used by some real-world data intermediaries as a way to enable more sophisticated AI applications that involve longitudinal patient journeys in order to drive timely patient recruiting. Thank you all very much.
First, I'd like to thank very much the FDA and the MHDS for inviting me for this exciting audience. For me, it's been very interesting to come to this because I'm not used to regulatory meetings. This is my first time dealing with this many regulators in a room, and I have to say it's been a very positive experience. I was a bit frightened that I'd want to run screaming from the room, but I didn't. I actually learned a lot about how it is that you make medicine actually apply to people, which I think is wonderful. So really what I wanted to do today was give you a sense of precision medicine at scale, which is really the idea of taking large amounts of data from different sources and using that to enable well-focused and precise treatments. So I wanted to start with a very old slide, actually. I pinched this from Dan von Hoff in 2016, and it's still current today. And it really lays out what the problem is that precision medicine is there to address. Fundamentally, the issue is that drug development is very wasteful. We're using a whole bunch of treatments where we actually don't really direct them very well. So let me just walk you through this diagram because it's a bit messy, right? So what you see there is a glimpse of all of the clinical trials in the year of 2016 that involved either a PD-1 inhibitor or a PD-L1 inhibitor. They're still the best cancer drugs that are out there. And what's interesting is that each one of those dots is a trial. The outer ring are the phase 1 trials that involve a few dozen patients. The middle ring is phase 2 trials that involve a few hundred patients. And the central zone is phase 3 trials that involve thousands of patients. Now, they're arranged out by different cancer types. And you'll notice that there's dots everywhere. The density of dots, the frequency of dots, has very little to do with the expected efficacy in each indication. It's everything to do with the supply of patients. And it turns out that in all of these boxes, less than half of the patients are responsive, in many cases even less than 20%. And so fundamentally what we're doing is recruiting by occurrence mostly. Too many patients aren't responding, and we're essentially screening in patients, which is a horrible waste of everyone's time and an enormous cost to everyone. So there is a better way, and this better way was actually pointed out by MD Anderson and others a long time ago. So this is from their website, but they had things like this up several decades ago. And the whole idea is that really the problem that we have with things like PD-1 inhibitors is that we don't understand why it is, what the different biologies are. That are, some biologies will be responsive to PD-1 inhibitors, others won't. And until we understand what kind of biology a particular patient has, you can't solve it. You can't address it properly. So the precision approach is very straightforward. You take this undifferentiated group of patients, you first study them with multimodal data, molecular profiling, or whatever else you can get, and use that to break them up into separate groups that have that represent the different biologies. You can then sort the patients into different biologies and hopefully find a drug to address each one of them. Using that approach, you can basically get much higher responses that are tailored for each patient. Now, in the background of this is the use of AI. So AI is extraordinarily helpful in doing something like precision medicine, because when you think about it, with precision medicine, you need to take large amounts of very heterogeneous data from many patients and figure out what's going on. As you're doing that, if you're trying to develop a precision approach towards a therapy, you also need to generate drugs. And this picture here is inspired by a graphic that I saw from Deep Knowledge Analytics some years ago, where they highlighted that in the standard sort of drug development flow of tasks, highlighted by the arrow there, basically AI and data science has a large role to play in contributing to all of the different stages, running from target ID through to clinical. And I stepped out a little bit more broadly and thought, well, after the drug's been approved, there's also post-market things that have to be done. You have to essentially optimize your manufacture. You have to sort of make sure that the market gets delivered properly. There's also repurposing opportunities. All of these things are highly enabled by AI methods to deal with all of the data that you need to do this. And I've highlighted examples of the kinds of data in the upper section. There's also the earlier stage for precision medicine, which maps out what MD Anderson pointed out, which is you need to understand your disease population and try to break them down into treatable groups. So what I wanted to do for this talk is just give you a view of how this has been done or is being done in different settings. And I'm just showing some settings that I'm familiar with because I was involved with them. The first is looking at translational oncology from work that I was doing around 2012. Then there's proactive medicine, when I was working with Google Verily to figure out how we can use multimodal data to enable essentially tailored treatments. And then finally, I'm going to spend most of my time talking about Owkin, the company that I'm currently affiliated with, who have built our whole approach towards treatment on a precision approach. And at the very end, I'd like to talk a little bit about the ecosystem of companies and players needed to make this happen, and a little bit about market trends. And hopefully I can do this all in 20 minutes. We'll see. So for starters, Novartis was actually a very early player in precision medicine. They basically, so Bill Sellers initiated a very grand mission to see whether or not we could understand cancer not as a number of individual diseases, but as one overall strategic view of all the different cancers that you can have. And in order to do that, they began this very ambitious project in collaboration with the Broad Institute called the Cell Line Encyclopedia. And the idea was to take all of the available cell lines that they could lay their hands on in the market, profile them with all of the most advanced profiling technologies, perturb them with gene knockouts and drugs, and essentially use all of that to try to characterize what might treat different kinds of cancers. You can get a picture of it of what that looks like here. So here is a slide that shows you some of the results coming from the CCLE. And so in addition to the CCLE data, which was all of these cell lines, we also internally ran about 1,300 drugs where we tested them at multiple doses across all of those cell lines. And the big chart on the left is showing you a clustered view of what all those compounds look like across all the cell lines, where green here means that you had a potent inhibition, and red means nothing exciting happened. And unsurprisingly, there's a whole bunch of drugs at the bottom where nothing exciting happens. There are a bunch of drugs at the top that are cytotoxic, where you basically knock out all the cells. Those tend to be actually pretty poisonous. And in the middle are the interesting ones that only affect some settings and not others. And those really are very promising for a precision approach. And so Novartis used that to, first of all, confirm that they could essentially predict the mechanisms that are needed for responsiveness, and then to essentially sort cell lines or disease types into groups that are more responsive to different classes of drugs. And they use that to basically enable their approach towards precision medicine. The other side of what they did was to also look at patient data. So Barbara Weber initiated a program where we would essentially take every single patient in our early clinical trials and have them genetically profiled at Foundation Medicine. So we could build out genetic profiles like what you see on this chart over here, where essentially these are the data corresponding to one PI3 kinase trial, where each of the patients on the left has a genetic profile that's laid out by the genes on the right. You can see mutations and copy number variants and things like that. And it basically allows you to understand what genetic features are associated with the responses that you can see in the PFS charts over there. So the whole idea was to build out hypotheses from the molecular profiling of cell lines, check that they work reasonably well in these patients, and then essentially have a way of addressing the patient's treatment in real time. We moved towards a rational patient allocation approach where we essentially were setting up adaptive basket trials, where we would take, and here's an example of a basket trial arranged around LGX818, which was one of our BRAF inhibitors at the time. And the idea basically was you take all these patients that you think are going to respond to your BRAF inhibitor. Some of them do and others don't. For the ones that do, that's fine. You keep them on the treatment. For the ones that don't, you then take a more careful look at their genetics, or maybe generate more, and use that to identify what would be a good combination therapy to combine LGX with another compound, and then send them on towards greater levels. And this led to actual trials that we initiated, and they executed well as far as I can tell. I left the company by the time this actually happened. So that's sort of one way of sort of going after it. Novartis was really able to do this because they were in an unusual position where they had a very large pipeline of many drugs. So by essentially running this trial, they had a good chance that when they found a treatment that would need to be done in a basket trial, they're likely to have a Novartis drug that they can send the patient to. And if not, they can send it to another company. The second section I want to talk about is some of the work that I was doing at Google Verily. And Google, in 2014, Google created Google Life Sciences, which became Verily. And the mission for Verily was to see whether we could collect an enormous amount of complicated data on a whole bunch of patients, combine that with cool technologies that monitor it in fun kinds of ways, create data systems that capture all of it, and then mix it all together and amazingly come up with novel insights. And the goal was really to move towards proactive precision medicine. Rather than waiting for a patient to become sick and then treat them, it would be nice to anticipate that they're about to come sick and treat them earlier. That was a very appropriate goal for Google in the sense that it was hopelessly ambitious, but we essentially had a chance to really do something very grand. So for example, one project that we began was the Baseline project, which at the time was actually a fairly straightforward idea. The idea was to take a set of patients. At the time that I was there, it was 10,000 participants. We were going to generate all of this multimodal data on them, so it was molecular profiling, imaging, and pulling together the medical records, revisit them every year for a period of 3 or 5 years, and at the end of the day, answer several basic questions. The first question is, do we see differences between the patients? And when we do, does that reflect something that might suggest a disease that we later on discover that they reached? We also worked at the other end of the idea, which is to essentially build image-based predictors of different conditions. And here's a really fun project that we initiated in collaboration with Mount Sinai, working with Eric Ehrman and others, where we essentially took Mount Sinai's collection of 37,000 head CT scans and came up with a predictor of whether or not it was an urgent case that needed to be immediately treated. And Eric basically built the model in a period of less than 6 months, took it back to the clinic, and demonstrated that it could actually shorten the decision process for patient triage by a factor of 3. Because too many patients already they were losing too many patients, because by the time they had a chance to review the scan, a patient might be too far along. But if you could basically accelerate those people to earlier in the queue, you could actually address them on time. And basically, this was a beautiful demonstration that this kind of method actually can save a lot of time. What I liked about this project is that it highlighted where AI is most effective, which is where you have a practical problem that relies on confusing big bits of data, and where you work very closely with the clinicians to define the problem, to measure the output, and deploy it. Now I want to move on to the Owkin bit. So coming back to the idea of precision medicine. So I showed you before the MD Anderson picture. Owkin, basically, our approach to precision medicine looks just like that. We take multimodal data, which in this case is clinical data, histology data, RNA sequencing, DNA sequencing of different kinds, and actually we're now moving a lot into spatial omic profiling as well, and single cell data. And we try to collect as much of that as we can on a whole bunch of patients. Once we've got that, we can then take a heterogeneous disease and break it into groups. So let me give you an example to make that clear. So when people receive a diagnosis of triple-negative breast cancer, you actually have a very bad prognosis. And the reason why is because there aren't very many approved drugs in TNBC. The reason why isn't because there aren't drugs that could work in TNBC, but it's because it's too heterogeneous a group. You actually have about 10 or 20 different biologies tucked into that diagnosis. And if you try to look for targets, it's really hard to see anything standing out. And if you do find a target that works in TNBC, your clinical trials fail because too few patients are responsive. So essentially, if you can break them down into clusters that appear to have separate biology, you can now look for targets that will address that biology and biomarkers that will help you assign patients to that biology, at which point you can really do precision medicine in the way that MD Anderson envisioned. So this slide sort of gives you a sense of what Owkin is doing at the moment. Actually, I think I missed one slide, but that's all right, that's good. So Owkin has built up a network of a large number of hospitals to gather a lot of patient data to allow us to do this kind of work. So it's interesting, our network is, I think, is very complementary to Rhino's. We have a lot of sites in Europe. We have a few sites in North America. We're expanding in North America as well. And the idea basically is to collect information from all of these samples to enable the collection of multimodal data. And like Rhino, we federate, and the reason why we federate is for all of those good reasons. It's very secure. It's compatible with GDPR, essential for us because we were building a network in Europe. It's very efficient, and the hospitals get to own and control their data, which is very important to them. And we have a couple of consortia that we set up along that way. Ittai kindly mentioned Melody, which is where we looked at drug screening data from different pharma companies. And we have a number of clinical collaborations where we're working with different institutes to generate federated data sets. And what we do with all those data is we put them into our engine. And our AI engines basically take the multimodal data that comes in from all the different patients, and we have 3 groups of computational engines that we apply. One group essentially is our biomarker engine, which looks for markers, visible, measurable markers that can indicate what biological class you might fall into in a subgroup. We have a discovery engine, which looks for targets that stand out in that subgroup. And then we also have our development engine, which looks for optimal ways to design and execute trials when you have better understanding of disease subgroups. This slide here gives you an idea of the kinds of models that we've been building in the diagnostic space. But so the left-hand side is one of my favorite models, which came from a recent publication called H2RNA. And what it does is it takes a standard pathology image, an H&E image, and from that image, it predicts the expression of about 200 genes at different locations within that tumor. The way it's able to do this is that it was trained on a data set that had paired H&E images and RNA-seq. And for about 200 of the genes, you could actually identify image features that corresponded to the RNA-seq. Since the image features are not uniformly distributed on the tumor, you end up having then a spatial distribution of each gene's expression. This is very exciting because to normally measure the spatial distribution of each gene is a very expensive, non-standard assay. And now we can infer it directly from a routine image that you already have. And we have a number of models that we built that all have that property in common. The whole idea is to predict something hard to measure from routine data that you already have. So we have ones that look at molecular state like this. We have others that look at disease outcome. And we have others that predict drug response. We're also trying to expand out our data in a new and interesting way. So we recognized about a year ago that we could really make major progress in the immuno-oncology space by collecting a lot of spatial omic data. So what do I mean by spatial omic data? There are new technologies emerging from companies like 10x and NanoStrain, where you can essentially measure the expression of thousands of genes in thousands of locations across a tumor. So you can directly measure the kind of thing that we were inferring in the H2RNA paper. The reason why this is helpful is because the tumor-immune interaction is highly locally regulated. So when you think about it, what is a tumor-immune cell interaction looking like? You have a bunch of immune cells drifting about in a tumor and deciding whether or not they want to kill the tumor cell. That decision is made very locally based on signals between the immune cells and between the tumor and the tumor cells. It's different in different parts of the tumors, and it's different in different patients. So using these kinds of data, you can basically take signals that would be very hard to detect in bulk across the whole tumor and understand what's happening in different parts of the biology at different times. These technologies came out a few years ago. Everyone realized they'd be amazing for this. But the problem is that they're very expensive and they're very hard to work with. So no one has run large studies up to now. The largest studies that we know of out there are basically a few dozen patients or so. So we decided to do thousands. And the way we can do that is by working with 5 major medical centers in our network. They're collecting the samples and generating the data on the premises, where 10x is actually giving us discounted access to their equipment. And so we can basically distribute the equipment at discounted levels. And then we are building a database that will allow us to all mine it together for immuno-oncology mechanisms that might explain non-response to PD-1 inhibitors, for example. So finally, what I wanted to do is just tell you a little bit about the ecosystem that is being built around what's needed for precision medicine, specifically in the AI space. So when you think about it, these slides really come from when I was in Big Pharma. I would have endless strategy discussions with people in Big Pharma about what data science was and what each company was. And you cannot believe how many people were confused about the difference between a company like PathAI and Recursion. They're very, very different companies, but not everyone had a clear idea. So I have here sort of a little roadmap that helps you make sense of this in an intuitive way. The most important difference between AI companies, I think, is what they're trying to sell you. So the ones on the left are selling you a model, the ones in the middle are selling you data, and the ones on the right are trying to sell you a pipeline. And essentially, you can differentiate many companies by understanding that space. Now, within that right-hand group, the ones that are trying to build a pipeline, which is where Owkin is pushing our major effort right now, they're also not all the same. They're also very different from each other. And the best way to make sense of them is to look at their capabilities. How do they go about finding a drug? So if you're going to find a drug with AI, you will essentially need to do the kinds of things that are mapped out in that chart, in those arrows on the top, where you'll have to have a data source to work with. You'll have to have a way to discover targets generally. You'll then need to have a way to experimentally validate your targets. And then ultimately, you need to discover a compound and run clinical trials. And the companies that used AI to discover drugs went about this actually with an interesting pattern historically. In the early days, on the bottom, there were the companies that first entered into AI discovery. They were very, very good at matching a novel molecule to a target that needs to be addressed. And there were a number of companies that came out looking at biologics like AbCellera and Moderna, and others were looking at small molecules like Schrödinger and Atomwise. And there's many, many others that have emerged in that space. Now, the next round of companies that emerged were the ones that got very clever at navigating the information that's out there to identify targets that you might want to address in this kind of a way. And they've essentially built out their capabilities along that strength. The next generation were in vitro and recursion, which came up with extremely clever phenotypes, which allow you to discover new targets that no one's known about before because of the novel phenotype that's revealed. And the top group is the one that Owkin wants to move into, is essentially the group that has access to large amounts of patient data. And that's the only one of these that can essentially map out the precision medicine approach by looking at subtypes within populations. And that's really where we see our big opportunity. So, speaking a little bit about trends, all of these companies are very nice, and if you're looking at it from an investor perspective, and I'm talking more and more with investors, you know, there's a lot of optimism about AI generally, but recently in the last year or so, a lot of pessimism about investing in biotech overall. And so I thought it was helpful to look at this little picture, because this picture makes two interesting points. It comes from a recent study that came out from, I think it was McKinsey, if I remember, and essentially, oh no, it's S&P. And essentially what this shows is that actually, as we had this latest dip in the market, biotech funding dropped dramatically. But what's interesting is that the number of deals didn't drop nearly as dramatically. So yes, there's a lot less money in biotech, but the number of deals is almost about the same. And the reason why is because rather than abandoning biotech investing overall, investors have basically shrunk what they're willing to spend on each one, and they've shifted their preference to companies that are generating revenue. So they're looking for proven track records and more lean investments to make. But if you're able to pull that off, you can actually still succeed as a biotech company and get funding. The other thing also is that if you look at the areas of precision medicine and AI/ML, those are still growing. So this is from a McKinsey study where they essentially mapped out how many deals are being made, and you can basically see that, you know, it's shrinking in a number of different sectors. But for the precision medicine field and for the AI discovery, there's more and more such deals being made right now in terms of investments from venture capital. On the right hand, and of course, it's even more extreme when you look at generative AI, where there's an enormous bubble of investing right now because it's a brand new, emerging, exciting field. So finally, I just wanted to end on a discussion of the ecosystem of all the different players that need to come in here. So I saw a very strong theme in this conference of saying how we all have to harmonize and collaborate and cooperate, and I couldn't agree with that more. And it's even more so when you think about trying to make precision medicine a real thing. And so the way I think about it is that there's sort of 4 different groups of actors in this field. There are, of course, the big pharma that generate an awful lot of drugs. There are the biotechs, like Owkin and many others, that are filling out discovery and development efforts in different ways. There are the academia, and the hospitals, who are handling all the patients and making all the hypotheses. And then there's venture investors. And the way to think about this collaboration is that each of these sectors has their strengths. So I've highlighted here a few strengths that they have. So what are big pharma good at? What they're really good at is late-stage development and manufacturing and distributing drugs. The others are hopeless at that. Pharma are amazing at that, right? The biotechs are really good at risky early drug development and for sort of putting into practice drug discovery methods. So that really early risky bit, they do extremely well. The other really nice thing about them is that when they fail, they go out of business and start another company. When a big pharma fails in a drug discovery effort, someone has to pay the bills. So they end up being much more risk averse. Academia is where all of the new ideas are going to come from. And it's not because pharma aren't brilliant or biotech aren't brilliant. When I was at Novartis, I was surrounded by brilliant scientists everywhere. But the fact of the matter is that even Novartis cannot possibly compete with the thousands of universities around the world, all of whom are discovering new biology. So the fact of the matter is that most of the new ideas are going to come from academia scattered across the world. And so how the collaboration needs to be done is essentially to think about the strengths that each of these sectors have and encourage the others to enable the collaborative enterprise that will make precision medicine happen. So what that means for pharma is they should be seeding R&D funding and projects that help develop biotechs and academic groups to feed in the way they can. Biotechs can innovate away and take the risks. The academics essentially can spit out ideas all the time, and venture can try to be clever about how to essentially invest in this space to encourage all of these different sectors. And that means particularly forming directed funds that will promote promising areas of research and promising business models. So with that, I just wanted to thank you very much, and sorry I went a bit over.
So hi, so my name is Hwiyoung Kim from Yonsei University, South Korea. So I would like to extend my gratitude for giving me this great opportunity to be here. And thanks for the great talks from the first day of the symposium, which was very insightful and interesting, so as I expected. But and thanks for the large screen and fancy podium, which was I didn't expect it that it is, but very makes me very nervous. But okay, I was a researcher in medical AI field, conducting studies on the diagnosis and prognosis prediction in the purpose of utilizing these techniques as a CDSS, clinical decision support system. So, and I'm involved in the foundational tasks aimed at applying these technologies in clinical settings. So with great sincerity, I intended to introduce these efforts in this talk. Okay, so I established the Translational AI Lab in Korean, 의료인공지능 연구실, to quickly meet the clinical unmet needs using artificial intelligence technology and to improve the standard of care. The purpose of AI-based medical data analysis goes beyond the simple identification of the biomarkers for screening and diagnosis. So as it aims to provide the patients with the opportunities for appropriate or optimized treatment and prevention, as well as to discover prognostic biomarkers that can offer accurate and consistent response assessment after treatment. So to achieve this, a variety of specific objectives was established. The first objective involved the identification of biomarkers for the diagnosis and prognosis prediction of cancers and various other diseases. So it's utilizing AI-based prediction models. So predominantly, our research efforts focused on the discovery of biomarkers through the analysis of medical images, which are representative phenotype of diseases. And ultimately, our goal was is to analyze the correlation between the macroscopic phenotype observed within the medical images and the cellular level phenotype observed in pathological images of patients, and along with their corresponding genotypes. So for instance, while a biopsy for histological confirmation is ideal for precise diagnosis, but brain tumors, which are difficult to biopsy, to have support efforts to extract as much information as possible from medical images. I too have collaborated with medical professionals at Johnson University to research technologies that employ AI to differentiate and evaluate the various subtypes of brain tumors using medical imaging data, something like this. So various subtypes was discovered can be done within the medical imaging analysis. And AI-based risk stratification is a solution that assists the healthcare professionals in making clinical decisions and enables appropriate treatment and prevention of patients. We have demonstrated the effectiveness of applying AI technology to predict the occurrence of the liver cancer in patients with hepatitis B who received the antiviral therapy based on their clinical data. And I would like to introduce a study analyzing gastric cancer by combining radiomic information extracted from medical imaging and genotype. Using AI-based analysis of gastric cancer images, we attempt to stratify the risk of the gastric cancer patients. Looking at the results, it observed that there was a significant differences between the low-risk group and high-risk group, only stratified by AI using medical imaging and clinical data. So when the same cutoff value was adapted, applied to the validation set, the significant difference was also observed. And after examining the added value of the AI system, based on stratification for adjuvant chemotherapy response prediction. It was found that when belonging to our low-risk group and the benefit group of previous studies based on the genotype, so published in Lancet Oncology, it is better to administer adjuvant chemotherapy. To contrast, in the high-risk group, it was observed that no significant differences in receiving chemotherapy, regardless of the belonging to the benefit group from previous study as a genotype. So, and we have developed and published an AI model that can predict which patients are at risk for osteoporosis without the need for medical imaging, specifically for the purpose of diagnosing osteoporosis. The model utilizes chest X-ray images obtained during routine health checkups. For this opportunistic screening of osteoporosis, this AI solution seamlessly operated in real clinic and helps to check potential health risk for patients, enabling proactive intervention by medical professionals. So our AI-based model demonstrated significantly better prediction performance than the existing rule-based prediction models based on known clinical data. And so recently, you know that as AI-based generative models like ChatGPT have gained significant attention, and they have also been actively explored in the context of biomedical and life sciences complex data. Notably, so efforts have been directed towards leveraging generative models, primarily led by generative observational networks, again, to address the challenges associated with the rare and complex condition like brain tumors. Sorry for the Korean words. So for brain tumors, where the available data is often limited, so making big data-driven research difficult. So we have aimed to tackle this issue of patient privacy concerns, which have posed obstacles to the practical utility of data, so particularly in terms of data usability. We have utilized the generative models to gain some synthesized brain tumor data for the big data study. And AI-based medical image synthesis technology demonstrates various applications. So for instance, we have developed a technology that decomposes interfering factors such as ribs in chest imaging to improve the interpretation of the images, so assisting the healthcare professionals in their diagnosis and clinical decision making. So as an example, we have developed and presented the technology utilizing AI-based chest imaging synthesis, which enhance the soft tissue visualization, which can aid in the diagnosis of lung cancer. And furthermore, it is possible to attempt the conversion between the different types of medical images. So with the help of AI technology, so we have developed technologies that infer CT images from MRI images acquired from actual patients. This technology provides additional information that can be obtained without the patient undergoing actual CT scan. So enabling more precise planning radiotherapy plan, such as gamma knife treatment. This technology has been commercialized. And the next research topic pertains to the utilization of the AI as a tool for biomedical research. So organoids have become a precise preclinical model for researchers for researching personalized drug development and organ-specific diseases. And optimizing our organoid culture, the condition requires periodic monitoring and precise interpretation by researchers, but thus researchers need an observant tool to understand the organoid images and evaluate its culture condition. To satisfy these unmet needs, we developed a deep learning-based organoid imaging analyzing algorithm, the organ extractor, organ structure is designed to overcome current obstacles of inefficiency to analyze the organoid images. And we developed AI-based model that performs real-time and end-to-end rodent behavior classification. So intended a tool for analyze rodent behavior experiments in specifically autism spectrum disorder research. And this model shows expert level human performance in distinguishing subtle behaviors associated with autism spectrum disorder. And such research in the field of medical AI heavily relies on medical data. To effectively utilize bioconvergent data, standardization is imperative. So furthermore, to ensure consistent validation and performance assessment of AI models based on this data, standardization should precede these efforts. This hierarchy of AIMD performance evaluation was presented yesterday by Jung from the AbbVie. We established the IEC 63521 standards, so machine learning enabled medical device performance evaluation process. So we need more attention and collaboration with various experts. Yeah. And so various efforts for clinical evidence of utilizing AI is performed as if AI replaces one of the radiologists, improved cancer detection rate was published on the Lancet Digital Health for the mammography screening tasks. Simultaneously, it demonstrates the potential to lower the recall rate for the patients visiting the hospital for additional examination. Yeah, this is some actual clinical evidence. And another evidence is for the segmentation tasks, which makes for the radiotherapy control planning. So automated radiotherapy control planning is needed for the consistent and accurate controlling for the specific disease sites, and also for the organal risk. So 3-year plan to gather more evidence on their use was established by NICE. And also, so we set up the comprehensive standard for AIMD. So in the IMDRF is actually serves as an arena for the international regulatory authorities to discuss harmonization in response to variations in regulatory standards across the different countries. So the standardization and regulatory standards for medical AI in South Korea are globally recognized as exemplary. But as a representative of South Korea, I had the honor of advocating and delivering a lecture at the annual conference in Sydney, Australia, emphasized the need for the comprehensive standardization framework for medical AI, which was like this, so from the data curation standard and labeling standard. So, and for the AI models, we set the reliable AIMD standards, which encompasses explainability, uncertainty, and fairness. And after the model development, the pre-market evaluation process and post-market evaluation, and also for the type of the medical device, ML in software, or ML as a software, we have to discuss the various types of the AIMDs, and we need this comprehensive standard, and so massive collaboration and discussion will be needed. And MFDS in South Korea actively participates the activities of the AI and machine learning working group of the IMDRF. So it engages in expert consultations to contribute to the development of good machine learning protest, GMLP, further development of GMLP, considering the characteristics of generative AI are underway. In summary, the AI for clinical decision support systems extends beyond the analysis of medical images for screening and diagnosis, so aiming to predict patients' prognosis, establish optimized treatment plans, and serves as a tool for evaluating treatment response. Generative AI also demonstrates various potential applications. However, further efforts are needed to ensure clinical effectiveness and safety, establishing standard methodologies for evaluating AIMD is necessary, and as well as implementing automated frameworks to facilitate how a flow of clinical data in clinic and real world is needed. And these efforts are essential not only for assessing AIMD, but also for post-market monitoring of AIMD. Thank you.
Thank you for the introduction, and good afternoon. I'm Jung from Sungkyunkwan University and Samsung Medical Center, and it's my pleasure to have opportunity to introduce current landscape of generative AI and its application in medicine. Let me start with the current landscape of medical generative AI. As you might be very familiar with these chatbots, with these chatbots, generative AI technology has been an integral part of our daily lives and changing the way we interact with computers from instruction to interaction. We can even create various forms of data, including image, audio, even video, using generative AI without a single line of programming. Based on recent breakthrough in generative AI technology and its potential in many industries, the landscape of generative AI in many domains is exploding by many startups and tech giants. And the market size of generative AI and generative AI in healthcare is also growing very fast, and we will see more growth in coming decades. So what's generative AI and what's special in generative AI? While most of the AI-based software as a medical devices are based on the discriminative model, which tries to find the decision boundary of diseases or disease severities, generative model tries to learn the distribution of input data set from which we can sample a new data set, even not present in the training set. As the distribution of the most of real-world data set, such as language, text, audio, video, images are very complex and high-dimensional, it has been very difficult to precisely learn the distribution of these data sets using conventional machine learning algorithms. However, as we have access to the large-scale training data set and we have now better algorithms using artificial neural networks or deep learning, we can train a generative model which converts given input to new data points which have similar distribution with the real-world data set. Another important aspect of modern large-scale generative AI is self-supervised learning training paradigm, which trains the model by utilizing the information inherent in the training data set without requiring expensive annotations. The contrastive learning is to make the model associate similar samples and dissociate dissimilar samples, while the generative pre-training tries to predict the masked or future input in the data set. As this self-supervised learning does not require explicit annotation, we can utilize internet-scale data set to build and develop bigger models. In the nearly a century of AI history, it's been only last decade since the deep learning techniques to train generative AI models have been actively developed. Even within last 10 years, many algorithms for generative models have been evolved from variational autoencoder generative adversarial networks to diffusion models and recurrent neural networks to transformers. So far, the most successful generative models for images would be generative adversarial networks and diffusion models. The generator of generative adversarial networks are trained to generate plausible and synthetic data, while the discriminator tries to distinguish the generator's fake data from real data. There are many interesting applications of generative adversarial nets in various fields, including medical imaging. For example, converting low-dose CT scans to normal-dose CT scans to reduce radiation exposure, or converting undersampled MRI to full-sampled MRI to reduce the acquisition time. And even we can convert MRI to CT scans to save the cost and reduce the potential patient harm. Recently, generative model called diffusion models has gained popularity for its capability of generating high-quality images, and has been the backbone of well-known image generation tools such as Midjourney or DALL-E. Diffusion models try to predict the noise in the corrupted image and progressively remove the noise to finally get the clean and desired image. While this iterative algorithm is more computationally demanding, diffusion models usually can generate more diverse and high-quality images than generative adversarial networks. Recently, diffusion models have actively applied to various medical domains, such as generating high-resolution chest X-rays in this slide, or generation of images with different phases or state for precise diagnosis or quantification. Another family of generative AI for mainly text or sequential data is recurrent neural net and transformers. While RNNs has shown many successful applications in many areas in natural language processing and speech recognition, it has limitations in dealing with long sequences. To this end, transformers was developed to handle longer context efficiently by using self-attention mechanism. Transformer consists of encoder and decoder part, which can be trained jointly or separately. Specifically, the decoder part, sometimes called GPT, is trained autoregressively to predict the next input or token given previous inputs. This decoder-only transformers has been the backbone of most of the well-known large language models, which is trained using internet-scale corpus of text and can be used to solve diverse natural language processing or understanding tasks such as translation, summarization, and most importantly, question answering and conversation. These generative AI models are not constrained to work in a specific domain. Generative AI can act as a domain translator, which converts a data in a domain to another domain beyond the text and images. We can develop this kind of multimodal generative AI using language as the anchor modality, or we can build a truly multimodal AI by jointly learn the representation of multimodal data in the same latent space. We can consider OpenAI's GPT-4 Vision and Gemini, Google's Gemini, as the early commercial level multimodal AI, which can understand combination of modalities and generate multimodal outputs. Now let me move on to the medical application of these recent progress of generative AIs. Maybe one of the most straightforward application of generative AI in medicine is to use LLM large language models for clinical question answering. In this study, ChatGPT has shown remarkable accuracy in answering radiology questions comparable to radiologists. And GPT-4 demonstrated significantly better performance than its previous version. In a recent study, various commercial and open source LLMs have been tested to answer nephrology questions, and again, GPT-4 significantly outperformed other LLMs. LLMs can also be used for facilitating informed consent for patients with various education levels. The informed consent forms are generally filled with many medical and legal jargons, and not all the patients have the reading proficiency to fully understand them. In a study recently published in NEJM AI, ChatGPT was used to simplify the consent form to middle school reading level. After the verification by a panel of legal and medical experts, the simplified forms turned out to be substantially easier to understand. This system has been already deployed in Rhode Island's largest healthcare system from last September. Using the speech recognition engine and LLM can be combined and integrated to EHR to develop an ambient agent which listens to and summarizes patient-clinician conversation and automatically generate clinical notes, such as Doc's Dragon ambient experience from Nuance. However, this kind of LLMs are reported to sometimes generate hallucinated information. To solve the hallucination issue of LLMs in medicine, a group of Stanford researchers developed retrieval-augmented clinical language models, which retrieves information from clinical knowledge base or search web to generate more factual and safe answers. Instead of using existing LLMs developed for general purpose tasks, Google developed MedPalm, a clinical question answering LLM fine-tuned with medical data. With the instruction prompt tuning for aligning LLMs to medical domain using few examples, MedPalm generated answers to clinical questions with the quality comparable to the clinician answers. The MedPalm tool, which used better-based LLM and novel prompting strategy, resulted in significant performance improvement, where the answers generated by the MedPalm tool were significantly favorable by the evaluators than the answers by clinicians with less potential risks. Google also recently tried to optimize MedPalm 2 for differential diagnosis to see the potential of LLMs as a diagnostic support systems. In the blind evaluation, the accuracy of clinician differential diagnosis assisted by LLM was better than the clinician diagnosis assisted by searching for web or reference book. And the performance was far better than the differential diagnosis by clinician alone. One interesting point in this study was that the differential diagnosis by LLM alone has demonstrated the best performance among all the settings. In medical image interpretation, one of the long-awaited application of generative AI would be the automatic generation of radiology reports. In this study, they developed an AI model which jointly trained image encoder, which embeds chest X-ray to image features. And the text decoder translates the image features into natural language in the radiology report form. In their test, the overall rating of the automatically generated reports were quite satisfactory that most of them didn't need any change or only minor changes were necessary. There are already a couple of commercial level products for automatic radiology report generation, while some are based on purely generative models, while some are combining existing and approved medical devices, software medical devices with LLMs. In the preliminary evaluation of study, radiology report generation AI produced significantly more acceptable quality reports than general proposed commercial LLMs such as GPT. Another application of generative AI in healthcare is to generate future health scenarios of individuals. By pre-training with large-scale electronic medical records, this transformer-based generative model can generate ICD code of future visits, which can be used for the prevention and management of each patient. In their evaluation, this generative model approach demonstrated significant improvement over existing models for predicting common or uncommon diseases. The final and the ultimate application of generative models in medicine would be building multimodal foundation models for generalized medical tasks. By using mechanism to facilitate the use of multimodal inputs, these large multimodal models, in short LMMs, can take multimodal interleaved data set as input and interactively answer various questions. Another LLM developed by Google called MedPalm-M has capability to digest various data sets from radiology, dermatology, pathology, and even genomics. Throughout the extensive evaluation published recently in AnyJM AI, MedPalm-M demonstrated better or comparable performance in various tasks compared to the best specialist model for each task. We will witness active development of these kind of LMMs in each domain of medicine in the coming years, just as the foundation LLMs for radiology in this slide to solve various radiology tasks. Let me conclude with regulatory and ethical considerations. As we are approaching to the moment of using generative AI in clinical setting, there are various risks and challenges we should consider at each stage of AI ML models development as a medical device. Oh, sorry. These considerations are mainly originated from the multimodal and generally proposed nature of generative AI. While there are some suggestions for the approval of generative AI, such as LLMs, we still don't have enough experience and consensus of potential risks and benefits of these AI models in clinical setting. However, we are not just sitting back and watch. These are the number of efforts to develop the guideline for safe and effective use of generative AI in medicine. AIRIS Symposium today is one of the very important events for this journey, and some international groups are working on establishing reporting guideline of LLMs in healthcare setting. WHO also has recently published really well-documented guidance on ethics and governance of LLMs for healthcare. Thanks to the hard work and collaboration of many stakeholders, including regulatory bodies such as FDA and MFDS, the field of AI ML SAMD has evolved at an incredible pace over the past decade. We now have hundreds of approved or cleared AI SAMD, both in the US and Korea. And we will see more of them considering the number of undergoing clinical trials. However, even though we are finally witnessing the adoption of AI SAMD is accelerating, the absolute pace of adoption is still slower than we hoped. This is partly because the discrepancy between what we expect to AI medical devices and what current AI medical devices can provide. The result is fragmented and duplicated MLMD landscape, and that's why there are many platform providers to combine and integrate these individual solutions. In the coming years in medicine, we will see more and more applications of generative AI and foundational AI technology that can solve a wide range of tasks by unleashing the potential of multimodal healthcare data. We are at a very interesting moment, and it's right time to discuss and design how we can best utilize this innovative technology to solve grand challenges we face, such as aging, clinician burnout, and implementing value-based care. Thank you for listening.
Hello, I just want to thank the organizers, both our Korean hosts and the FDA, for inviting me and giving me a chance to talk to you about causal AI and learning biology at scale. We heard a little bit earlier about how there were so many research labs out producing great work, advancing biology, but we have a long way to go, and I want to give you an example of where we want to end up. So, if we're looking at a computer chip here, this is a circuit diagram for a transmitter. This is a digital twin of a transmitter. This has all the information that you need to know to make a prediction about any possible intervention on the system. And if you are an electrical engineer, this is second nature. This is just the world that you live in. Well, we aspire to have models that can tell us about the biology at that level of detail, where the model can tell us what the impact of an intervention is, to tell us what we will see when we do the experiment. And we do have genetic circuitry. We can we have models that look very much like circuit diagrams, but the problem is 95% of the circuitry is missing. So even if every edge and node in that network is correct, it's not going to produce very good answers about what will happen when we make an intervention that we've never seen before. That's why we rely very heavily on experimental techniques to go in and intervene, and we do randomized trials to make predictions about what will happen, because we don't understand the circuitry. So what would it look like if we could understand the circuitry? We could make detailed predictions about experiments that haven't been done yet. Well, what do we need to do that? So we need causal learning. A lot of the tools that we talked about this week are about correlation. So they tell you, what will I see? What will happen? What will I see if I see these characteristics? If I'm looking at an image, can I classify it as there cancer in the lung or not? They will not tell you what will happen when you make an intervention. And so we didn't need a different kind of math that answers causal questions. So what happens when I do? The other thing that we need, because there's not enough knowledge yet in the literature, is we need data. So we need multimodal data. This data is emerging. We've had lots of discussions this week about the detailed nature of this data, how you don't just have gene expression anymore, you have spatially resolved gene expression, you have single-cell gene expression, and all these layers describe the biology in detail, going from DNA, RNA-seq, proteomics, the clinical EMR, patient characteristics, and the treatments and outcomes. We work with a number of partners that provide the data. We don't do it ourselves. And we often work with partners that provide disease-specific data sets. So what do we do with these multi-omic data sets? We layer them together to build data frames that describe all the observations that we have. So these are typically not from clinical trials. They can be, but typically it's real-world observational data, clinical genomics data, that describes in great detail the state of patients when they were treated and ultimately their outcomes. The other piece that we need to do this is causal AI. And causal AI is built off of math that was developed by Judea Pearl in the late 90s, early 2000s. He won the Turing Award in 2011 on this math. The Turing Award is essentially the Nobel Prize in artificial intelligence. And we have taken this math and scaled it to work on the size of data that you'd see over on the left. How do we do this? We have a platform called REFS. We are engineers and physicists, so we're not that creative in naming things. So REFS stands for reverse engineering and forward simulation. We start by taking all the biological building blocks, all the variables in those modalities, and putting them together into small models, local models that describe how a few variables come together, and enumerate all the potential ways that those variables could come together. That's a relatively easy problem, even if it involves trillions of network fragments or puzzle pieces. The hard part is putting those pieces together into a global model, a generative model of all of the data. So we're not just looking to describe the patterns in the data. What we really want is a model that describes the biology that created the data. So we're reverse engineering the biology. That's an NP-hard problem. We use Metropolis Monte Carlo to walk through the space of all models that are consistent with that data, and that space is vast. So a typical model, there may be, given the data that we put into it, more potential models, more potential biologies, as Joseph said earlier, than there are atoms in the universe. And so we have this enormous problem to solve to find the models that are consistent with that data. And once we've learned those models, we don't learn a model, we talked about uncertainty and risk. We learn a distribution of models. We sample from that model space. And so to make sense of that, we run synthetic experiments against those models and get a distribution of answers out. So we have self-consistent error bars. We know how much we know. We know what the uncertainty around any specific question that we ask. So this is all a little bit abstract. Let me get a little bit more concrete. So here's a typical example. So this was a data set from G3 Global Genomics in cardiac. This was a prospective trial design, so no randomized treatment, but there was a very controlled set of how patients were brought in, about equal numbers of cases and controls. And in those patients, whole genome sequence was measured, RNA-seq, proteomics, metabolomics, lipidomics, and about 500 of the classic biomarkers that you'd want to measure if you're interested in coronary artery disease. We took all that data and put it into a data frame where the columns represent the observations on each patient. Each observation is a row. So that's simple. You can put small versions of this in Excel, but it's just those constraints on what was observed. And now we're going to try to reverse engineer what the biology is that gave rise to those observations. So we do the enumeration, we try all the puzzle pieces in all the different ways that these variables could come together. Could it be a DNA variable driving RNA? It could be RNA driving an outcome, all of those possibilities. And we do the optimization and sampling to find the sample of networks that are consistent with the data. And what comes out is very much like what you see on the right. A graphical model that contains some known biology. The black arrows represent what's known. The red arrows represent novel biology that's never been seen before. So this is not coming out of a database. This is not coming from mining the literature for relationships. These relationships, both the black and the red, are learned ab initio up out of the data. And so when we find things that are known, that's confirmation that we're on the right track. And these models represent different versions of biology that are consistent with the data. The more the data we have, the cleaner the data we have, the better the measurement technologies are, the more these networks all resemble each other. But they have everything from the DNA to the RNA transcriptomics out to the molecular phenotypes, the disease outcomes that we want to modify. So the kinds of questions that we can ask. So at Aitia now, we're very much focused on drug discovery. And so what we want to do is distinguish between the two cases over on the left. So all of these variables are correlated with each other. If you did differential gene expression, everything here would be, all these variables light up. But only some of them are on the causal path to the outcome. So if I intervene on the left and I follow the signal down to a phenotype, when I do that synthetic intervention, so imagine it's an siRNA knockdown, we're doing a synthetic one on all the models, we get a distribution of results. A good target looks like the result on the left, where intervening on that variable, on that modality, leads to a change in the phenotype. Another variable on the right that's correlated, but not causal for the endpoint, there's no movement in those distributions. We can also do drug simulation, where if you have an idea of what your target is, you're already in clinical development, we can tell you what the predictive biomarkers are for that target. What are the right patients? Okay. So here we have a number of cases where we have validation to various degrees of certainty. We work in neurology and Alzheimer's, in oncology, in multiple myeloma. I'm going to show you an example today from cardiovascular disease where we discovered a novel receptor. But we also have cases that we can talk about in drug simulation. And please find me afterwards if there's any of these specific cases you'd like to discuss. So I'm going to talk about coronary artery disease, a model that we built with G3. And there's two key players in this model that are really interesting. So triglyceride-rich LDL particles. These are little spheres of lipids containing cholesterol on the inside, and this is how cholesterol is transported. And also LP(a) is another kind of particle that's very interesting because it is the most, there's the most heritability in this. So you inherit 2 copies from your parents, and depending on exactly which form you've inherited, you have a differential risk for coronary artery disease. So this is what the data looked like. So even in controls and cases, working with G3, we had wonderful partners who understand the disease deeply. This is not all about the math. You have to understand the disease biology. So we had very strong quantitative measures of the clinical state of the patients through CT, whole genome sequencing, RNA transcripts, the proteome, metabolomics, and lipidomics. And so we had, this is the number of data points that we had on any single individual in this study. So we took that to get that, and we said, all right, well, let's build a model. So this is roughly the structure of the model. So clinical parameters like age, and maybe BMI, the DNA variants pulled from that set of 1 billion measures on each patient's DNA. And then each of the modalities, DNA methylation, microRNA, messenger RNA, proteomics, metabolomics, lipidomics, and the biomarkers, these all could interact. We said, this is really the only hypothesis that we put in, is that DNA drives these layers, and those layers drive the clinical phenotype. Everything else was hypothesis-free, and we have to learn the structure from the data. So to do that, the data frame ended up having about 100,000 variables after all the hard work of data cleaning. This was built on about 300 patients. So that's really small. That's not very, we are well underdetermined when you think about the number of parameters in these models we're learning. Our training sets are not millions of pages from the internet. They're 300 patients with good quality data. We do the reverse engineering during enumeration. We looked at about 10 to the 18th network fragments. I don't know how to pronounce that number, the number of networks that are possible given these constraints and the data that we used. But we sampled about 10 to the 11 networks in learning the model. Once we have the model, then we do synthetic experiments on it. We like to characterize the model by intervening in every single variable in the model and measuring the response of every other variable to every one of those interventions. And so those pairwise causal simulations, there were about 10 to the 10 of those that we did. This all took about a week to do. So, and the amount of computing, so we improve our algorithms probably about a factor of 2 to a factor of 10 every couple of years. And so our scientists just use that, they make the models bigger, more detailed, more resolution, but in a reasonable amount of time, we can reverse engineer the biology behind a disease and learn that biology at scale. So this is the entire disease process, not just what's happening around a single receptor. So, how am I doing? So I'm going to show two stories. So the one about the LDL subnetwork, this was published in a paper last January in Frontiers in Cardiovascular Medicine. So when we reverse engineered the model, the model had 100,000 components in it. This is just a neighborhood in the vicinity of coronary artery disease. And you see a lot of the familiar players. So CRP, small LDLs, ApoAB, triglycerides, and it turns out that in the models that we learned, that LDL triglycerides were the variable that was most causal for the coronary artery disease outcome down at the bottom. This is not from an experiment that measured that. This is reverse engineered from all of that biology. So we can look in more detail around this. So if we look at the lipase C gene, the DNA, so we go back to the DNA layer, we can see that SNPs and lipase C modulate the expression level of lipase C. We don't have the protein, so we're missing that modality. I'd love to have it. But we can also see that those variations drive differences in LDL triglycerides that also show up in differences between case and control and probability that you have coronary artery disease. So we can look in detail at the mechanisms. The models give us the path from the DNA all the way through to the phenotypes of the disease. And very quickly, I'm going to give you another story around a novel receptor. So this is another look at the neighborhood around of the network that was learned for coronary artery disease. Just grossly at the top, you can see there's DNA variables in the purple at the top. So RNA are the green variables, blue are protein, and down at the bottom is coronary artery disease. So we learned these are the mechanisms that go from genotype through to expression to outcome. If we look in detail, what we found was when we look at Lp(a) metabolism, so it turns out that there are two forms of Lp(a). Lp(a) has ApoA protein that wraps around the outside, and it has a repeat in it called a kringle, and you might inherit a short version of it that has a small number of kringle repeats, or a large version that has a long kringle repeats. And so nobody knew exactly what receptors process this, and it turns out that when we look at the model, we can see that the Lp(a), the short version of it, the K1, is processed by the LDL receptor. We didn't take this out of the literature. This is directly the topology that was learned by refs. It turns out the large version, the long version, when the number of crinkle repeats covers the ligand for the LDL receptor, it's cleared by a different enzyme that was not known to be involved in Lp(a). I can't tell you what it is because there's efforts to develop drugs against this target, but I'm going to show you the experimental validation. So the model directly learned new biology that can inform treatments for patients. So when we look at, there's a specific molecule, the model predicts a specific molecule, the long version of Lp(a), with a specific receptor. Now we can go and see if there's additional evidence for it. So if we look at the genetics for that receptor, we go to that receptor and look at all the variation across our population, and when we associate that with the levels of large Lp(a), we get a significant hit. So that circled hit. So we see variation. We found evidence that there's variation in the gene of that specific receptor and the circulating levels of large Lp(a), but not, and then as a negative control, but not small Lp(a). So those p-values are not very small, right? So if this was GWAS, If this was a genome-wide association study, that would be just in the noise. But because the network told us where to look, it's this receptor. It's here where the biology is interesting. This is now strong enough evidence for us to be able to have confidence in the result and go forward with an experiment. Okay, I've got 11 seconds. Okay, so this is a case where we have in vitro validation. So a biopharma partner took that receptor and developed an siRNA knockdown against it. And as a control on the left, you can see the gray is the negative control where there's no knockdown. The hatched on the right is the siRNA knockdown, and as a positive control, you can see that the known substrate of the LDL receptor, its uptake is downweighted with the siRNA. But also, when you knock down the siRNA of the novel receptor, you also change the uptake of Lp(a). So this is experimental validation that the biology that we discovered using causal AI to discover new biology is actually validated. So I've got another case, but it doesn't look like we have time for it, so I'm going to end here. But I want to thank you guys again. This was I'm really excited to be here and give this talk.